welcome to Physics Wala's brand new NEET English YouTube channel that's made especially for you. Let me quickly introduce myself. I'm Shalini Somashekar and I've been teaching botany for the last almost 15 years now. And in today's lecture, we are going to be covering the chapter Cell the Unit of Life in one shot. I'm sure as NEET aspirants and biology enthusiasts, you're already aware that cells are tiny microscopic structures that living organisms are composed of, right? Despite their extremely small size, cells are capable of doing so many phenomenal things. In fact, they act like tiny factories within which they have their own recycling units, they have their own powerhouses, they have their own security systems, all about which we're going to cover in today's lecture. And in order to make your lives easier, your preparation easier, I'm going to be sharing the lecture notes along with some questions for practice on the app, which you can access for free using the link given in the description box. Now, while you're on the app, you might as well explore the app, because on the app, you're going to find a lot of quizzes, structured resources that's going to help you get closer to your dream of becoming a doctor. Now, without any further ado, let's get today's session started. Now, before I actually deep dive into the chapter Cell the Unit of Life, I want to give you a clarity on two things. Number one, how many questions can you expect from this chapter in the upcoming NEET exam? If you carefully analyze the last few years' NEET exam papers, you will notice how every year a minimum of 3 questions have appeared from this chapter, and there have been times where as many as 6 questions have been asked from this chapter alone. So this chapter clearly carries a lot of weightage. It's not a very difficult chapter. It's easy to understand. I will try my best to make it easy for you to understand, and also it's not really complicated. If you pay 100% undivided attention throughout the course of this session, I'm sure you'll be able to answer all of those questions in the NEET exam. Now, the second thing is whether you should refer the old NCERT textbook or the new updated one, the latest version for this chapter. I would suggest everybody to refer the newest, the most recent NCERT textbook for this chapter, because there have been a couple of places where the information is slightly changed right now and updated right now. It's best that you read the updated information. Now, if you are somebody who attempted NEET exam in 2025, I'm sure you'll be traumatized by that one question which was about reductionist biology. There was so much chaos and confusion surrounding that one question. Many people believed that it was out of syllabus, not mentioned in NCERT. But turns out that is a term that is very much given in the introductory part of this very unit. So we've learned our lesson from NEET 2025. We cannot skip any little line or word from NCERT. We'll cover the introductory part, we'll cover the summary, and we'll cover every single line, every single word mentioned in the NCERT textbook. All right. So this chapter is one of the chapters in the unit. In fact, it is the first chapter of the unit, Cell, Structure and Functions. In this unit, you have 3 chapters. The first one is this one, of course, and the second one is called Biomolecules, about which you will learn in the zoology syllabus. And the third chapter is called Cell Cycle and Cell Division, which we will learn in botany itself eventually after a couple of lectures. And yeah, this is an introduction to the entire unit. So this unit introduction begins with the definition of biology. What is biology? Everybody already knows. Biology is that branch of science that deals with life and living organisms, right? So biology as a field of science has seen a lot of change, has evolved with time. Initially, when people started to get interested about living organisms around then, microscopy had not evolved that much. We had not known about the kind of molecules, the kind of chemicals that living organisms are composed of. We did not know about molecular biology. So back then, the field of biology was mostly descriptive, right? So they looked at organisms and they tried to describe them in simple terms, morphological features. Like if somebody who likes or is interested in biology comes across a unusual looking type of plant, they would start describing like, this plant has a root system of this kind, it has leaves of this kind, the leaves are arranged in this way, the flower is of this color. Oh, this giraffe, it has a very long neck. This porcupine, it has spines on its body. The elephant has really long fan-like ears and a long trunk. So most of the initial part of the study in biology back then was descriptive. So as and when people started describing living organisms, they figured about the enormous diversity there was in the living world. Of course, many species, many different characteristic features exist, and there's a lot of variety in living organisms. Not all living organisms look alike, right? So they were kind of mind blown about the fact that there is so much diversity, so much variety in the living world. Also, what intrigued them was the fact that despite being so diverse, There could be an animal like a hippopotamus. There could be an animal like a lizard that could climb on the wall. There could be an animal like a snake that crawls on the ground. There could be burrowing animals. There could be different types of plants. But regardless of this enormous diversity that we see in the living world, there is something that unites us all. All living organisms are composed of cells. Despite the diversity, there is a unifying factor that every living organism is composed of cells. And a couple of years later, sometime later, something called the cell theory got proposed. So, more about the cell theory we will explore in this chapter. Once the cell theory got created, basically it says that every living organism is composed of cells. There was a sense of mystery around living phenomena. There's a lot of physiological processes, behavioral processes that take place in living organisms. What people started to notice was, unless the cell was intact, unless the cell maintained its integrity as NCERT describes it, many of these phenomena could not be observed. Unless a cell is maintaining its structural organization, it's having all of its components organized well, many of these biological phenomena would not occur. Okay, so they figured out that cell is very, very essential. The cell has to remain as a unit, remain as in that fixed organization for all of these processes to happen. Now they were curious about how these biological processes or these physiological phenomena take place inside of cells, take place inside of living organisms. They wanted to find out how any process works. So the approach that they took was the physico-chemical approach, which is called reductionist biology. Basically, it's like, imagine you're somebody who is very much intrigued about how a clock works. By just looking at the clock for an entire day, you're not going to figure out how it works, right? In order to, kind of understand how is it that the second needle will move, how is it that the needles inside a clock are spinning, what are the parts that are found inside the clock, what is making them move, how does the battery inside work. Unless you break things down, look at its inside, you will not be able to figure out how any structure or any phenomenon works by just looking at it from the outside. So then what people started doing is they started reducing the phenomenon into small chunks. Okay, so basically they broke the cells open. They tried to understand what are the kind of biomolecules that are present inside, what is the biochemistry of the cell. Once they started figuring out, okay, this tissue has these kind of chemicals, this tissue or this cell is made up of these type of proteins, then the question was, okay, this protein is present in the cell. What exactly is it trying to do inside the cell? What function is it performing? How is it performing it? So in order to understand that, by just studying about the exterior structure or just the morphology won't suffice. For that, you'll have to break things down, which is exactly the approach, which is reductionist biology. Okay. So eventually they started finding out about how molecules interact with each other, how are certain chemical reactions giving rise to certain processes, and all of that. In order to do that, they also made use of what we call cell-free systems. As the name itself indicates, a cell-free system is a system which is composed of living components, but the entire structure of a cell is not intact. For example, even without an entire cell, it's possible for us to create or synthesize proteins in the lab. You need ribosomes, you need amino acids, you need an mRNA template and a couple of enzymes. Basically, even if the entire cell is not there, some biological phenomena can be demonstrated in laboratory settings. They started analyzing what chemicals are present inside the cell. By making use of cell-free systems, they kind of started to understand how these different chemicals interact with each other and give rise to biological phenomena. In the chapter Biomolecules, you will learn about what are the different kinds of organic molecules and organic substances that you find inside living organisms. Okay, so this chapter mostly focuses on how cells structure is, what are the various functions of the components you find inside of a cell and all of that. Okay. Now, in your NCERT textbook, in the beginning itself, there is an activity that is given where you're asked to identify the difference between living and non-living things. Right now, I have a couple of pictures here on the screen for you. And among these pictures, I want you to pick the ones that are living. There's pictures of a plant, there's a couple of pens, some books, a cute little puppy, there's a fish, there's soil, there's sunlight, and there's water. I'm sure it should take you less than 3 seconds to pick the pictures which are made up of living organisms. Of course, a plant has life, this cutie puppy has life, and this fish has life. So do many, many microorganisms that we cannot see with our naked eye, right? So all of these are living as opposed to sunlight, wind, water, soil, and all of that. They don't have life. Now the question is, what is it that these organisms have in them that makes them living? What do we have that non-living things don't have? I already mentioned, right? Even though there is so much diversity, these two don't look alike at all. This is a plant, an entirely different kind of organism. This is a puppy, an animal, an entirely different kind of organism. There's so much difference in the way these two organisms appear. But regardless of all of that diversity, they are alike in one feature, which is they are composed of cells. Every living organism is composed of cells. So we have living cells that give us, make sure that we are surviving, we are alive. But soil and sunlight and water and temperature and air, all of these are non-living. Okay, so there are no cells in them. Okay, so cells are what make living organisms living. Now, if I ask you about what is a cell, as biology students, I'm sure this is not the first time you're hearing the term cell. You've been reading about cell, I think, at least from 8th grade, right? Regardless of which state board or CBSE board you're from, you would have been introduced to the concept of cells in living organisms very early on. And I'm sure your teachers must have already told you that even your lower grade, cell is very important. The definition of cell is important. You must memorize it. So even if I come in the middle of the night, probably at 3 a.m. and wake you up and be like, define cell, you will even in your sleep say that cell is the basic structural and functional unit of life. Everybody knows about the definition. But have you ever really sat down and pondered about why is it that we call the cell the fundamental structural and functional unit of life? Why can't an organelle, which is smaller than a cell, be called the structural and functional unit of life? Why can't something bigger than a cell, like a tissue, be called the basic or fundamental structural and functional unit of life? Why can't we do that? There are few reasons for that. Now, if you look at living organisms, there are organisms that are composed of just one cell. Amoeba, bacteria, paramecium, many protists, all of them are composed of just one cell. Their entire organism itself is made up of one cell. As opposed to that, you have many, many multicellular organisms, including us, many animals, many advanced plants. They are multicellular. Now, as multicellular organisms, there are certain living processes that take place inside of us. Like we all respire. Cellular respiration is a characteristic feature of living organisms. In order for us to survive, cellular respiration needs to happen. Various other physiological processes take place inside living organisms, such as reproduction, such as excretion, such as digestion of food. The complex physiological processes, life processes that take place inside of us multicellular organisms also take place inside of cells. An individual cell in unicellular organism is capable of all of those functions. In amoeba, you see digestion. In amoeba, you see reproduction. In amoeba, you see cellular respiration. In amoeba, you also see, did I mention reproduction? I probably did. Movement, that is one of the features. Of course, it's not the characteristic feature, but they also display signs of life. They are also showing life processes. Now, if you think about the various levels of biological organization, the smallest unit that is capable of displaying all signs of life, all the life processes, is a cell. If you take something smaller than a cell, like an organelle, can an organelle perform all of those functions? Can an organelle exist independently? Now, these cells, amoeba, bacteria, other protozoans and protists that I'm talking about, they have independent existence. Even as a single cell, they are displaying all signs of life despite being a single cell. Can something smaller than that do it? Can an endoplasmic reticulum alone, you detach that, take it out of a cell, can an endoplasmic reticulum alone survive like a living organism? It cannot. Can a nucleus, which is considered to be the smartest part of the cell, like the brain of the cell, you take out the nucleus, just the nucleus, and keep it as one living organism, can it function? It cannot function. So, the cell is the smallest structure which can exist independently, which displays all the life processes that multicellular organisms show. Anything smaller than a cell, other smaller organelles that you find inside the cell, is not capable of existing independently. So the smallest unit that can exist independently, performs all the functions like multicellular organisms, that is the basic functional unit of life. Now, why do we call cells the basic structural unit of life? Again, all living organisms, regardless of our big size, are composed of tiny cells inside. It's like a house may not, it could be any big, it could be like a multi-storied building, but still the house is made up of bricks, right? Structurally, what is the house made up of? It's not like one block of something. You have tiny, tiny, tiny bricks that are put together to make this huge building. Similarly, no matter the size, it could be a blue whale, it could be an elephant, it could be a mammoth, it could be us. Regardless of how big we are composed of many, many tiny, tiny cells. Our structure is made up of cells. So cells are the basic structural as well as functional unit of living organisms. Okay. So unicellular organisms are capable of existing independently, and also they do perform all the essential functions of life. Anything that's less than a complete cell is not capable of existing independently, which is why we call cell the basic or the fundamental, structural as well as functional unit of living organisms. Now, how did we know that cells exist? The first person to observe living cells under the microscope was Anton von Leeuwenhoek. And the first person to discover the nucleus inside a cell was Robert Brown. Every little detail is important. The names of scientists, what is their contribution, everything is important. Questions can appear from anywhere, okay? So don't assume that contributions of scientists is not important. I suggest you make separate notes, maintain separate notes just for specifics like this. Names of scientists, names of, contribution of scientists, in which year was some research published, in which year did something get discovered. So all of these specific details that you have in NCERT, it's best that you make a list, a separate notes for that so that you can revise it frequently. Okay. Now, I told you about the cell theory very briefly when we started this chapter. The cell theory is a theory that was proposed by two people and later one other person contributed to it. He modified it a little bit and then it was published. Okay. So the first person to have contributed to the cell theory, his name is Matthias Schleiden. He was a botanist from Germany. This is Matthias Schleiden. In the year 1838, just around the time when microscopy was, kind of getting advanced. Being a botanist, of course, he is interested and intrigued by how plants are. He observed many, many different types of plants that grew around him. He took different parts of different plants, the root of plant, stem of plant, leaf of plant, and he started observing them under the microscope. And he noticed that regardless of the plant, regardless of the part of the plant, all plants were composed of cells, different kinds of cells, and these different kinds of cells form tissues of the plant. Now you might think, is this even worthwhile to study about? What kind of a contribution is this? This is so insignificant. But back then, to us it is insignificant today because microscopy is advanced so much. These are things that we know just generally like that. Anybody will know this. But back then, when people had not really looked inside of living organisms to see what kind of structures we are made up of, back then, this was very, very significant. He for the first time said that living organisms, especially plants, regardless of which plant, which part, all of them are composed of many different types of cells, and these cells combine to form tissues inside the plants. Now, another person at around the same time in 1839, his name is Theodor Schwann. This Theodor Schwann was a zoologist. I mentioned how in this year's NCERT edition, there is a change, right? One of the changes is that in the earlier versions, Theodor Schwann is mentioned to be a British zoologist. Right now, they've changed it, changed his nationality. Apparently, he's German. So, from British, it is changed to German. So, this is important. Okay, so this is in the updated NCERT textbook. So, he is a German zoologist. Obviously, a zoologist will be interested about animals. And he was also trying to kind of get to know more about the internal structure of animals and plants. And along with, looking at animal tissues under the microscope, he also started to look at the tissues of plants under the microscope. So when he studied both plant tissues as well as animal tissues, he of course agreed that living organisms, be it plants or animals, are composed of cells. But when he observed plant cells and animal cells, he saw one striking difference between plant cells and animal cells. Around plant cells, there was a very distinct cell wall which was absent in animal cells. In animal cells, the outermost boundary was the plasma membrane. The plant cells, of course, had plasma membrane too, but that was not the outermost boundary. Surrounding the plasma membrane, the cell wall was also present. He notices that, he mentions that difference, and he also says that in both plants and animals, you find cells and its products. Living organisms, both plants and animals, are composed of cells and its products. He hypothesized that bodies of animals as well as plants are composed of cells and products of cells. Now, what do we mean by products of cells? What is product here? Product can mean two things. Something that the cell is producing. Probably the cell is producing certain chemicals and it's secreting them. That is a product of the cell. Also, cells can come together to form a tissue that is also product of the cell. Basically, what are we? We are cells. Our body has whatever the cells produce. Our body has tissues that the cells are made up of, right? I mean, that are made up of cells. So, he says that both plants and animals have cells and products of cells. He reported that cells had a thin outer layer called the plasma membrane, and plant cells have a cell wall which animal cells did not have. Fair. Now, there was one question that was kind of unanswered. When multicellular organisms grow, we increase in size. The number of cells in our body increases. It's not like we were one cell and then that cell increased in size and we became this fat, right? We started off as a single celled zygote, but the cell divided and divided and divided and gave rise to this multicellular body, which is today composed of trillions of cells. How did that come about? How did that one cell end up giving rise to so many different cells? If new cells have to form inside the body, where is it forming from? So this is something that the both of them did not address, and this was addressed by another scientist by the name Rudolf Virchow in the year 1855, much later. So he says, if in a living organism growth is happening, newer cells are getting added, it cannot just appear from anywhere, from thin air like that. You can't have some hocus pocus abracadabra magic and make new cells appear inside a body. In order for an organism to, develop new cells as we grow, the existing cells will have to divide. Only when the already existing cells undergo cell division, one cell becomes two, two become four, and in that way the number of cells in our body will increase. Otherwise, there is no increase in, the number of cells. New cells can just not arise from anywhere. This he describes in Latin as omnis cellula acellula, which simply translates to the fact that new cells can only arise from pre-existing cells. It cannot just appear out of nowhere like that. Okay? So he includes this statement also in the cell theory, and he kind of modifies the cell theory. And today we know cell theory in the form of these two statements. The first one being, all organisms, including plants and animals, are composed of cells and the product of cells. The second statement is, all cells arise from pre-existing cells, which means cells that are already present, only they can give rise to newer cells. Okay? Now, you've learnt about two different types of cells, plant cells and animal cells, in your lower grades. If you're a CBSE student, probably, if I remember it correctly, you would have learnt about it in 9th grade. You would have learnt about plant cells using the example of a cell from the peel of onion. For an animal cell, you would have used the example of the human cheek cell. So this is just like a quick recap to relate to what you had studied in your lower grades. Clearly, onion peel cells have a very distinct cell wall. These are plant cells. Of course, there is a distinct a cell wall around plant cells, which is absent in animal cells. Both plant cells and animal cells have a plasma membrane, and they have a very distinct nucleus inside every cell, which you can see in the form of these dots here. Every cell is made up of a very distinct, prominent nucleus. Now, you can classify cells into two types based on one criterion. Okay, of course, you can classify cells into many different types. One of the ways in which you can classify cells is based on whether the genetic material in that cell is covered by a nuclear membrane or not. Okay. Now, based on the presence of nuclear membrane, cells can be classified into two types. Okay. The first one is called a prokaryotic cell and the second one is called a eukaryotic cell. Over here, pro means primitive and eu means true. What is karyotic? You might wonder. The term karyotic is derived from the word karyon, which means nucleus. If the cell has a primitive kind of nucleus, then we call it a prokaryotic cell. If a cell has a true nucleus, it is called a eukaryotic cell. What makes a cell a true nucleus? I mean, a eukaryotic cell, or what does it mean when we say it has a true nucleus? What we basically mean to say is the genetic material is covered by a nuclear membrane. If the nuclear membrane is present, if the nucleus is bound by a membrane, then we call it eukaryotic. If the nuclear membrane is absent, genetic material is not bound by a membrane, then we call it a prokaryotic cell. Over here, you can see this bacterial cell, a classic example for a prokaryotic cell. You can find there is DNA lying here in the cytoplasm. Okay. So, this DNA, as you can see, the genetic material is not covered by any membrane. There is no membrane surrounding it, right? So, it is not bound by a membrane. Whereas this eukaryotic cell here, an example animal cell, there is DNA inside, but surrounding the DNA, you have a very well defined nuclear membrane. This is the nucleus opened up. Okay, so a section, you can see DNA inside and there is a nuclear membrane surrounding the DNA. So the presence or absence of nuclear membrane is the basis for dividing cells into two types, prokaryotic and eukaryotic. Now, of course, the nuclear membrane present in one type and absent in another type. What is common to them both is the presence of a cytoplasm within the cell. The cytoplasm is a semi-fluid which is mostly composed of water. It occupies the maximum space, maximum volume inside the cell, and that is where a lot of metabolic activities and cellular reactions will take place. And it is the activities that take place in the cytoplasm that are mostly responsible for keeping us in a living state, for making sure that we are alive. Okay, so that's something that is common between these two. Now, is the absence of nuclear membrane the only difference between the prokaryotic and eukaryotic cell? I want you to carefully observe the two cells. This cell seems very empty. This cell is very crowded. There's a lot of structures that are present inside this cell. Another difference between prokaryotic and eukaryotic cells is that not only do prokaryotes lack a membrane-bound nucleus, they even lack other membrane-bound organelles such as endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and a whole lot of other organelles that are membrane-bound. There is a lack of membrane around the genetic material. Also, they lack membrane-bound organelles such as ER, Golgi, Mitochondria, Lysosomes. As opposed to that, eukaryotic cells do have many different types of membrane-bound organelles, which are mentioned right here. More about each of them we will learn as we, move into the depths of this chapter. So now just understand that this is another difference. Now I'm mentioning that this prokaryotic cell has no membrane-bound organelles. Does that mean it does not have any organelle at all? Not really. It does not have organelles that are bound by membranes, but it has non-membrane bound organelles such as ribosomes. Ribosomes are not bound by a membrane. They are present in prokaryotic cells and they are also present in eukaryotic cells. since they are found in both type of cells, ribosomes are also called universal organelles. Okay, so you can find ribosomes here inside the cell. You can also find ribosomes here. What is their structure? What is their function? What do they do? All about that we will learn as we move on. Now, let's talk about the size and shape of different types of cells. I mentioned how we are multicellular organisms. We have so many different types of cells. If you've learnt about animal tissues in your lower grades, you will know that we're made of many different types of tissues. There's connective tissue, epithelial tissue, neural tissue, muscular tissue. Likewise, even in plants, you have parenchyma, collenchyma, sclerenchyma, xylem, phloem, and a lot of that. Okay. So the cells in these tissues are not alike. The cell in squamous epithelium is not like the cell in columnar epithelium or in a connective tissue or in your bone or in your blood. It's not the same. The size is not the same. The shape is also variable. Compare your neuron, which is a cell in your own body, with your squamous epithelium lining your alveoli in the lungs. Are they the same in shape? Are they the same in size? Not really. Even though we are made up of many different types of cells that arose from a single zygote, they are differentiated. They would have modified themselves according to the function that they are meant to perform. Today we will look at some variations in the size of cells and also the shape of cells. Now, prokaryotic cells, the primitive type of cells, they are usually smaller compared to eukaryotic cells. There is a type of prokaryotic cell called mycoplasma, which is the smallest known living cell. It measures around 0.3 microns in diameter. I think micron itself is super tiny. In 1 millimeter, if you make like 1000 equal units, probably 1 unit will be equal to 1 micron. So it's not even 1 micron, it's less than that, it's 0.3 micron. That's how tiny this one particular cell is, or this particular organism is. If you take other bacteria, their length can measure between 3 to 5 microns. And when you consider eukaryotic cells like a red blood cell, red blood corpuscle, or erythrocyte, usually that will be much larger than these prokaryotic cells, and its diameter will be around 7 micron roughly. Okay. Now, in our body, some of the longest cells that exist are the neurons. There is a neuron that starts from the base of your spine, okay, and it moves all the way till the toe, the big toe in your feet. In very tall individuals, the length of that one neuron could be as long as 1 meter. Some of our cells, I'm telling you, are microscopic. You can't even see them. But there are cells in our body that are extremely long as well. So it's not like every cell should measure this much size only. So you see variety and variation in the size of cells. Here is an NCERT illustration giving you different ranges of size that you see in different types of cells, which I want you to take a look at and memorize. Now, talking about the shapes of cells. I mentioned how the cell size is not the same everywhere. The cell shape is also not the same. You know how in squamous epithelium, the cells are like super flat. They call it pavement epithelium. It's present in capillaries. It's present in the lining of your alveoli. Why is it so? Because across those cells, exchange of respiratory gases need to happen. If it's like one big fat cell there, exchange will take forever. There is too much resistance. In order to make that exchange or transport of respiratory gases easier across the membranes, the cells are like super flat there. You can find some cells that are disc-like this RBC, it is disc-like biconcave. Why is RBC biconcave? Why can't it be like one spherical cell? Why is it supposed to be biconcave? The reason is basically because of the kind of job that RCB has taken up. I'm a Bangalorean and I tend to say RCB a lot. So even if I by mistake say RBC, I mean RCB. Even if by mistake I say RCB, I mean RBC. Okay. So here, these are red blood cells, and these cells are biconcave in shape. When they mature, they lose their nucleus and become almost kind of flat. Okay. The reason they are of that shape is because of the kind of job it does. The RBCs are majorly involved in the transport of respiratory gases within our body. you take in oxygen, it goes to your lungs, from your alveoli it goes into your capillaries, and the blood is supposed to transport that oxygen to every one of our cells. So within this RBC, there are hemoglobin molecules, a kind of multi-subunit protein that will bind to oxygen and go transport it to every one of our living cells. Okay. In order for RBC to efficiently do its job, a lot of hemoglobin molecules are required. If there is one big fat nucleus sitting in the center of the cell, it will not be able to accommodate many hemoglobin molecules. So it loses its nucleus, makes space for a lot of hemoglobin molecules so that it can do its job very efficiently. The job is of transporting respiratory gases. Another reason why being biconcave and being small helps it is if it has to deliver oxygen to our tissues. Our tissues need oxygen because oxygen is what will help in generating energy out of food, right? So without oxygen, our cells cannot generate energy, they will die. So near the tissues, if the blood has to go and deliver oxygen, if RBCs have to go and deliver oxygen, they have to squeeze in through super tiny capillaries. If they are very rigid and very fat cells, they will not be able to squeeze in through those capillaries and deliver oxygen. So in order to, they take their job like it is the most important thing in the world, the cells, and they will do anything to make sure that that job happens efficiently. So because of the kind of function that this cell is supposed to do, it is shaped that way. What about white blood cells? White blood cells are very important cells in our body. They are what protects us from disease-causing harmful microbes and pathogens. They are part of our immune system, right? Let's say you were riding your cycle, playing cricket, playing badminton, dancing, doing whatever, you accidentally fall on the ground and then you get hurt. So when you fall on the ground, there is a skin barrier is broken. Bacteria are everywhere around us, on the soil, in the air. It's possible that because your skin is broken right now, it's torn right now, it's damaged right now, the microbes that are present around you can gain entry into your body. Once they gain entry into your body, they can cause infection. So that is possible. Your WBCs are so smart, they will identify what cells belong to us, what cells don't belong to us. The moment they notice that some cell that is not part of my body has entered my body, dishum, they will start destroying it and neutralizing those microbes. So in order to do that, ours is a closed circulatory system. So blood is usually getting transported through the blood vessels. These guys will have to get out of the blood vessel, go to that site of infection and start fighting all of those bacteria and pathogens. So how do they do that? In order to do that, they will have to squeeze through the spaces in the capillaries and go to the site of infection. If they're very rigid, they cannot do it. That's why they are amoeboid. They can squeeze through the spaces in the capillary wall in the squamous epithelium and reach the site. If they are rigid, they can't do that. That's why they are amoeboid. Talking about columnar epithelial cells, in cells like in the small intestine where a lot of absorption is happening, or in cells like in your respiratory tract where substances will have to be trapped, like harmful substances, dust particles, pathogens will have to be trapped, you find these kind of cells. The reason they are columnar is because if the cell is long, it gives it more space for a lot of organelles. It can accommodate a lot of organelles, and at the apex it can get modified. It can either have cilia like it does in the respiratory tract, or it can have villi, which will increase the surface area for absorption, microvilli, as in the case of your small intestine. So that way, that will give it provision to get modified. And talking about neurons, neurons are this crazy, super cute cells that have this unique hairstyle and a very long tail. The reason neurons are the way they are is because they are involved in transferring information from one part of the body to another. If there are many dendrites, branches like that from the cell body, it can receive information from a lot of neurons simultaneously. And because of the long tail, it can pass on that information very quickly to wherever it needs to be passed on to. Okay. And then some of these cells are extremely long, like tracheids and fibers. They allow, the shape of that cells allow it to be stacked one on top of another for providing mechanical support or for transport. So basically, the way they are shaped is because of the kind of function that they're going to perform inside the plant or inside the organism. Mesophyll cells are round and oval. Usually you have these polygonal cells or spherical cells. Why they are shaped that way is because whenever there are irregular spaces inside the organism's body, these kind of cells will fill up those spaces very easily. That is why if they are filling spaces inside the body, generally they tend to be polygonal in shape. Okay. So the reason why the cells are shaped the way they are is because of the kind of jobs that they perform inside the body of the organism. Now, coming back to prokaryotic and eukaryotic cells, in this chapter we'll have to discuss in detail about prokaryotic cells and also about eukaryotic cells, what prokaryotic cells are composed of, how is their structure, what organelles do you find there, and all of that. So right now let's get started with prokaryotic cells, and eventually we will learn about the eukaryotic cells in detail as well. So which cells are prokaryotic? All the cells that lack a membrane around the genetic material, that lack a membrane bound nucleus, are said to be prokaryotic. Bacteria, mycoplasma, cyanobacteria come under this group. Okay, fungi, protozoans are all eukaryotic. Only bacteria and mycoplasma, of course, that will also come under bacteria only. All of these are prokaryotic. So blue-green algae, there's another name for blue-green algae. They are also called cyanobacteria, which I'm writing in cyan color, blue-green color, right? So all of these lack a membrane around the nucleus or around the genetic material. Mycoplasma is also called PPLO, which is short for pleuropneumonia-like organism. They are known to cause respiratory disorders like pneumonia in certain animals. Okay, so that is why the name. Okay, now, yeah. In comparison with eukaryotes, like I already mentioned, these cells are much smaller, but when it comes to division and how quickly, how rapidly they multiply and increase in number, the prokaryotic cells divide more frequently compared to eukaryotic cells. So one cell becomes two cells, two becomes four, their number increases much faster compared to eukaryotic cells. Talking about its shape and size, like many other cells, this also varies widely in its shape and size. But you can find bacteria in four major shapes, which is what is mentioned here. It can be spherical. When it is spherical, we call them cocci. Cocci is the plural term. Coccus is singular. You may have heard of streptococcus, staphylococcus. Coccus is each cell is spherical in nature. If it is rod shaped, we call it bacillus. Bacillus is singular and bacilli is plural. You've heard of lactobacillus, Bacillus thuringiensis, all of them are rod-shaped bacteria. Then you have spiral bacteria, helical bacteria that are called spirilla, plural, spirillum, singular. You've heard of Helicobacter pylori that causes infections in the digestive tract in the stomach, right? So that is a spiral bacterium. If it is comma shaped, it is called Vibrio. Vibrio is plural, Vibrium is singular. Can you guess an example? It is a pathogen that causes cholera in humans, Vibrio cholerae. These examples are not mentioned in your textbook. Just so you relate, I'm giving you these examples. Okay? Now, yeah, here is a bacterial cell. A bacterial cell has its cell envelope. The most important thing in a bacterial cell that differentiates it from a eukaryotic cell is that around the genetic material there is no nuclear membrane here. The genetic material, even though there is no nuclear membrane, you shouldn't assume that the DNA in this cell is freely floating inside, swimming inside like it is in a pool. Even though there is no membrane around it, where the genomic DNA exists inside the bacterial cell, that place is localized. That place where the genomic DNA exists is called the nucleoid. It always is in this region. It's not like it's floating around and swimming around inside the cell. So, in bacteria, you find one chromosomal DNA, or this is also called the genomic DNA. This genomic DNA is circular What? Circular? This doesn't look circular in any way if you're wondering. It's actually a circle. It's like DNA is like this here. Okay? What I mean when I say it's circular is that there are no free ends. In eukaryotic DNA, it is linear. There are free ends present in DNA. Here there is no free end. But that big circle to exist inside the cell is a little difficult unless it becomes condensed. So what it does is think of it like a rubber band. If you're, folding that rubber band, what happens is what has happened here. How exactly it gets condensed and all of that, we'll learn in the upcoming chapters. For now, you don't worry about that. So this is the genomic DNA or chromosomal DNA. The genomic DNA or chromosomal DNA per bacterium, there is always only one. One in number. Now, apart from the genomic DNA, extra chromosomal DNA can exist in bacterial cells. It's not like it will 100% be there. It may be there. Okay. Now, this is absolutely essential for the functioning of a bacteria. Genomic DNA, chromosomal DNA, 100% has to be there. But in addition to this, in some bacteria, you can find circular DNA that we call plasmids. Plastids and plasmids sound very similar. These are not plastids, they are plasmids. Now, what are these plasmids? These plasmids are extra chromosomal, which means they are present in addition to genomic DNA. Just with this, bacterial cells can survive, but without this, with just this, they cannot survive. Okay? So, even if this is not there, these plasmids are not there, bacteria can still survive. But if plasmids are there, that will give extra advantage to bacterial cells. How? I'll tell you. Okay? So, these are extra chromosomal circular DNA. Its number need not always be 1. It can be 1 to many. When they have to make more copies of themselves, they don't rely on chromosomal DNA. They can make more copies of themselves by themselves. They have their own origin of replication. So they can replicate by themselves. Replicate means making an extra copy. So they can self-replicate. Sometimes these plasmids can have antibiotic resistance genes. What do antibiotic resistance genes mean? Antibiotic is poison to bacteria. If you give bacteria antibiotic, it should die, right? It should not be able to grow anymore. But if there are certain antibiotic resistance genes present in bacteria, even if you give it poison, it'll be like, oh, I don't care, I'm going to live my life like that, okay? So the presence of plasmids that contain antibiotic resistance genes are a boon, are a blessing to bacteria because antibiotics cannot touch them then. Also, sometimes plasmids have genes that will allow conjugation, a kind of sexual reproduction, a primitive type of sexual reproduction that you see in bacteria, more about which we'll learn as we move on. And yeah, that's about plasmids. If they're not there, bacteria can still survive, but if they're there, definitely there will be an added benefit to the bacteria. And these plasmids are extensively used in the field of biotechnology to introduce new genes into bacteria and stuff, more about which you will learn in the biotechnology chapters in zoology. Okay? Now, let's talk about the structure of the bacterial cell starting from the outside. The outside of the bacterial cell is called the cell envelope. The cell envelope is made up of 3 distinct layers. There are 3 layers present, but collectively as a unit, they function to protect the cell. A wall in any structure is meant to provide shape and protection, right? What does the wall in your house do? It gives a structure to your house. It protects you from the outside wind, sunlight, animals, and all of that, right? Same thing even for bacteria. So the wall or the cell envelope is going to provide protection as well as provide shape to the bacteria. I mentioned how the cell envelope is composed of 3 layers. Let's see what those 3 layers are. The first one, the outermost layer, is called the glycocalyx. The second layer inside the glycocalyx is called the middle cell wall. Inside the cell wall, you have what is called the plasma membrane. Plasma membrane is common to all living organisms. Cell wall plants also have, but structurally what it is made of will differ in bacteria, plant cells and fungi. Okay, and you have a glycocalyx layer. Now let's see what each of them are composed of. If you look at this, particular bacterial cell, the outermost layer, which is labeled in this bluish gray sort of a color, that is the glycocalyx. The glycocalyx is made up of polysaccharides. But which polysaccharide is it made up of? That varies from one species to another. What it is composed of is polysaccharide. Which polysaccharide is variable. Also, what is variable is how thick this glycocalyx is. How thick is that layer? It can sometimes be super thick or it can be very thin. When it is thin, you call it a slime layer. When it is thick, you call it a capsule. Okay? What is the difference? Apart from this being thin and this being thick, there are other differences as well. When it is thin, the slime layer is not very tightly attached to the cell wall. It is loosely just adhering to the cell wall. So it's very easy to wash it off. But when it is thick capsule, it attaches or it is very tightly bound to the cell wall. So it's very difficult to wash it. The capsule can give the property of virulence to bacteria. Okay. So basically it can make the bacteria cause disease in other organisms. Like for example, there is one strain Streptococcus pneumoniae bacteria that can cause disease in mice. But the strain without the capsule cannot cause disease. Simply because sometimes the chemicals in the capsule mimic the chemicals present in the host. So the host's immune system will not be able to identify them as a foreign object. Also because of its slimy nature, if any phagocytic cells like macrophages or natural killer cells try to engulf it, it will slip away. Okay. So the immune system may not be able to recognize these cells, which is why they will successfully parasitize the host cell and cause infection. Okay. Now, yeah. So let's talk about the cell wall. That's about the glycocalyx. In most prokaryotic cells, the cell wall is surrounded by the cell membrane is surrounded by a cell wall. Cell wall is present in most prokaryotic cells. It is absent in mycoplasma. Important. Mycoplasma lack a cell wall. They only have the cell membrane similar to animal cells, but they are prokaryotic still because they lack a membrane-bound nucleus. Now, what is a cell wall made up of? It is made up of polysaccharides and peptides. Basically, it is called peptidoglycan, or sometimes it is also called murine. That is what the cell wall is composed of. What is the function of the cell wall? The cell wall is supposed to provide shape to the cell, like I already mentioned, right? It provides shape to the cell. It provides a strong structural support to the cell and make sure that the cell does not collapse, right? That is what it's meant to do. Now, what about the cell membrane or the plasma membrane? The plasma membrane structure in prokaryotic cells and eukaryotic cells is almost very similar. When we discuss eukaryotic cells, we learn about the structure of the plasma membrane in absolute detail. For now, we will not deal with it. We will learn about it later. So now just understand that the cell membrane or the plasma membrane, or some people also call it plasma lemma, that is a selectively permeable membrane. It does not allow anything and everything to pass through it. So there's a lot of checkpoints in the cell membrane where selectively certain molecules are only allowed to pass into and out of the cell. Okay, so it is through the cell membrane that the cell interacts with its outside world. How exactly all of that happens, we will learn when we are studying about the structure of cell membrane in eukaryotes. Now, with respect to the various layers present in the cell envelope of bacteria, you have two different types of bacteria. One type of bacterium is called gram-positive bacterium. The other type of bacterium is called gram-negative bacterium. Over here, the gram is not about how much weight bacteria has, kilogram, milligram, gram. That is not the gram here. The gram here refers to the name of a researcher, of a scientist called Christian Gram. He devised a staining technique to specifically stain bacteria. Staining technique means coloring bacteria. If you have to look at their cells, if you have to visualize the structures inside the bacterial cell, you have to add color to them and look at them under the microscope, right? So that is called the staining process. There are many different ways in which you can stain bacterial cells or any cells for that matter. One of the ways in which it is done is called gram staining, that was devised by this person called Christian Gram. So when that gram staining procedure is done, at the end of gram staining, some bacteria will retain a blue color stain, some bacteria will become pinkish in color. So depending upon whether it will retain the blue stain or become pink color, you call it gram positive or gram negative. I'll tell you about that technique shortly. So now just understand the difference in the structure of cell envelope of gram positive and gram negative bacteria. Here we will not talk about the glycocalyx, we will talk only about the cell wall and the cell membrane. In gram positive bacteria, you have a cell wall that is made up of peptidoglycan, which is very thick. In this case, it is represented in these purple color. Peptidoglycan is like a mesh. Okay. So you have glycan, sugars, polysaccharides, cross-linked with peptides. Okay, small amino acids. So it forms a mesh-like structure. You have several such nets lying one on top of another, and you have many layers of peptidoglycan in gram-positive bacteria. As opposed to that, in gram-negative bacteria, you have very few layers of peptidoglycan. So it is very thin here. The cell wall is very thin. Here it is represented in cyan color. And inside the cell wall, obviously, you will have the plasma membrane, similar plasma membrane in both cases. Now, I want you to observe what is present outside the cell wall in gram-negative bacteria. Along with a plasma membrane lying inside the cell wall, they have an additional membrane-like structure very similar to the plasma membrane lying outside the cell wall. This is called outer membrane, and this outer membrane is present only in gram-negative bacteria and not in gram-positive bacteria. It is because of this structural difference in the cell envelope or cell wall of bacteria that they respond to the same staining technique differently. So what exactly does he do in gram staining? This is additional information. It's not there in your textbook, but I feel like you should know it. So I'm adding this here. There are 4 steps. First, you take bacteria. You don't know whether they are positive, gram negative. You add one primary stain, which is called crystal violet. Crystal violet, as the name itself indicates, is violet or bluish in color. So once you add crystal violet to the cells, I told you that the peptidoglycan cell wall is thick, right? Is like a net. And basically this stain will move into the cell wall, into the plasma membrane and go inside the cell, and all of the cells will get stained blue in color. Both gram positive and gram negative will become blue in color. Next, what they do is they will add iodine. Iodine is a mordant. Iodine will also go inside the cell. Iodine will bind to the crystal violet molecules and make it more stable. Now the crystal violet molecules will not be able to leak out of the cell. They are kind of fixed there. Okay. Then what they do is they will add a solvent like acetone or alcohol. In gram positive bacteria, what happens is even though the alcohol is added, because iodine is there, the crystal violet does not leak out of the cell. But in gram negative bacteria, when you add alcohol, the outer membrane is there, right? It is mostly made up of lipids. That will get dissolved. It will break down. And they have a very thin peptidoglycan layer through which the alcohol goes inside. It will decolorize the crystal violet. The crystal violet will it will leak out of the cell. After this, they will add a counter stain, which is called safranin, which is somewhat pinkish red in color, you can say. Once safranin is added to gram positive and gram negative cells, the safranin will go inside both the cells. In crystal violet, in the gram positive cells, since crystal violet color is still there, it will still dominate, and even though safranin is there, its color will not show. Whereas in gram negative bacteria, since the crystal violet stain has leaked out of the cell, they will start appearing pink or reddish in color. So after this whole 4 steps, if you look at the cells under the microscope, if they are appearing blue in color, that means they are gram positive. If they are appearing red or pinkish in color, that means they are gram negative. Okay. So, this is gram staining. Depending upon which color it is at the end of the process, you call it gram positive and gram negative. Why positive, negative? This crystal violet stain is also called gram stain. Whichever retains the gram stain is positive, so gram staining. This one has leaked out the gram stain, so this is gram negative. Okay, so that's how it is named. Now, let's talk about some of the structures that are associated with the plasma membrane in bacterial cells. I told you how bacterial cells do not have any membrane-bound organelles like mitochondria. If it's photosynthetic, it does not have plastids. It does not have lysosome, Golgi, ER, nothing. All it has is one plasma membrane. Now, all cells require energy to survive. All cells require to undergo cell division in order to grow, right? And if a cell has to survive, it needs energy. And where do we get energy from? From ATP. Where is ATP in large quantities produced inside of our cells? It is produced in the mitochondria, which is called the powerhouse of the cell for a reason. These guys don't have a mitochondria, but they're surviving. How do they get their ATP from? So what if they don't have mitochondria? They have plasma membrane, they will do some jugaad with that only and manage with that. Okay. So if you look at a mitochondrion, which of course is absent here, you have the inner membrane which is enfolded into cristae. The reason this is capable of producing so many ATPs is because the enzyme that is required for ATP synthesis is present here in the inner membrane. It's located here, right? So if the inner membrane has infoldings like that, it increases the surface area, it can accommodate a lot of those enzymes, and it can produce more ATP. Unfortunately, bacterial cells do not have such organelles. What do they have instead? They have an outer cell wall, which I will just label it as cell CW for convenience. Inside that, there will be the cell membrane. This is the cell membrane. The cell membrane structure is very similar to the structure of the mitochondrial membrane. Similar to how the mitochondrial inner membrane forms infoldings, the cell membrane itself will form infoldings, okay, like that. Sometimes it forms elongated tubular structures, sometimes it forms rounded spherical structures, and sometimes it forms sheet-like structures. And by folding upon itself, it will increase the surface area. So it is within these regions that the enzymes required for ATP synthesis are located. Even though it does not have an exclusive structure for cellular respiration, it makes do with whatever it has. Okay. So here, these structures, infoldings of the plasma membrane in bacteria are referred to as mesosomes. Okay. So it is a special membranous structure. How is it formed? It is formed by the extension of plasma membrane into the cell. Plasma membrane is extending into the cell towards the cytoplasm. They can be in the form of vesicles, tubules, and lamellae. This is a vesicle. This is a tubule. It's like a long tube, so it's like a tubule. This is a vesicle. This is sheet-like, so it is called lamellae. Okay, it can exist in any of those forms. Basically, it helps in cellular respiration, expansion of the membrane, increasing the surface area, secretion of substances, generating energy, and all of that. Okay. Also, they help in formation of a cell wall. Whenever bacterial cells divide and they have to form a cell wall between the two new cells after a nucleus has divided, these are the sites at which the cell plate formation usually begins. DNA replication and distribution to the daughter cells. Once DNA has made an extra copy, there are certain regions at which the DNA is kind of attached to the plasma membrane. That way they will help distribute that extra copy of DNA to the daughter cells that will form. Like I mentioned already, it helps in respiration because it has enzymes required for synthesis of ATP. It helps in secretion because there's increased surface area. Secretory processes also can take place. Yeah, basically it does all of that. But actually, there has been so much research in the last few years which have almost concluded that mesosomes are not real structures inside bacterial cells that you consistently see in all bacterial cells. Apparently, in one kind of fixation of bacteria on slides, whenever they are making like permanent slides, they will add cells on the slide and they will fix it, right? So during that fixation process, to look at bacterial cells under the electron microscope, sometimes apparently very harsh chemicals are used. And because of that chemicals, in response to those chemicals, the plasma membrane will take up like weird shapes. It will bend in weird ways, and that is called, that they assume to be called as mesosomes. But apparently, if you use many other different ways of, fixing these cells on on a slide, which are using less harmful or less harsh chemicals, you don't apparently find mesosomes. So it's not like a structure that exists in every bacterial cell. So most people say that they're just artifacts, like while producing or preparing slides of bacteria for electron microscopy using those chemicals only, then you see them, otherwise you don't see them. There is no special need for mesosomes as structures to exist. The plasma membrane itself can do all the jobs that the mesosomes do. So they're not like real structures, but NCERT has not updated that information. For your exam, this is important, I've told you. And as a teacher, as a science enthusiast, biology enthusiast myself, I also have to let you know and update you about what research has been going on in these fields in the last few years. So I've done both parts. Okay, so learn this as it is for your exams. Now, you know how many bacteria are photosynthetic? Like there are purple sulfur bacteria, green sulfur bacteria, cyanobacteria that are capable of performing photosynthesis. If an organism has to perform photosynthesis, then it requires sunlight. It requires a mechanism by which it can trap sunlight. It should absorb the energy from sunlight and use that energy to synthesize food. That's what should happen, right? So what helps in absorbing energy? Absorbing solar energy specifically. It is the pigments. In plants, you have many different types of pigments like chlorophyll, carotenoids present that will trap the sun's energy and use it to synthesize food. And those pigments in higher organisms are located in the membranes of the thylakoids inside the chloroplast. But here, unfortunately, poor little beings, they don't have a chloroplast, but they still have to perform photosynthesis. How do they do that? They use whatever they have, the same jugaad. There is a cell wall. Okay, so there is a cell wall. Inside the cell wall, there is the cell membrane. The cell membrane itself will fold upon itself into the cytoplasm and form structures called chromatophores. Chroma means color. Okay, chromatophores. It is within these parts of the membrane that pigments responsible for photosynthesis are located. This is not an artifact. This is a real structure that you can consistently see in all photosynthetic bacteria. Unlike mesosomes, which may or may not exist, this will exist consistently in bacterial cells that are photosynthetic. Okay, so this is found in some prokaryotes like cyanobacteria. They are membranous extensions into the cytoplasm and they contain pigments. Now let's move on and learn about some extensions from the cell envelope like the flagella, like the fimbriae and pili. Let's talk about flagella. Flagella are locomotory structures. You can find flagella even in eukaryotes. Functionally, they're both the same. They both help in moving the organism forward or just help in its locomotion. But structurally, eukaryotic and prokaryotic flagella differ very much. Okay. So here, bacterial cells may or may not have flagella. It's not like it's a constant feature that you see in every bacteria. Some bacteria are capable of locomotion. They can move actively and some bacteria cannot. In bacteria that are capable of moving by themselves using energy, actively moving, flagella is the locomotory structure. Okay, if the bacteria is non-motile, flagella will be absent. Now, how many flagella will be present? What is the position? How are they arranged in the bacteria is variable. Some bacteria may have only one flagella on one side. Some of them may have two on either side. Some may have a tuft of flagella on one side. Some may have two tufts on either side, and some it may be present throughout the surface. So how many are present, how it is arranged is variable. Okay, but structurally all of them have the same structure of flagella in prokaryotes. A flagella typically is composed of three parts: a basal body, a hook and a filament. From inside to outside, basal body is the bottommost part of the flagellum which passes through the membrane. Okay, so it's present inside the cell. Okay, so here this part is the basal body. This part that arises right outside the membrane of the cell or the right outside the envelope, that is called the hook. It is kind of flexible. And the longest part of the flagellum, which is completely outside the bacterial cell envelope is called the filament. Okay. So the filament is, oh my god, that's such a bad diagram or line. Let me make this hook over where there's sufficient space for our flagella. I'm sorry, for the filament. Okay. These are the three parts. And flagella in bacteria is composed of a protein called flagellin. Now, there are also structures, other appendages that arise out of the cell envelope in bacteria which may or may not be present, again similar to flagella. These are called pili or pili. I don't know how it's actually pronounced. That's how I've learned it from my teachers. Pili are plural form. Pili is the plural form. The singular form is called pilus. Not piles, pilus. Singular is pilus. This is plural form. These are elongated tube-like structures. Notice it is tube-like. It is like empty inside. There is space inside. It's like a tube. Tube-like structures that are made up of a protein called pilin. Flagella, flagellin, pilus, pilin. Okay. Now what is its function? It will help in a primitive type of sexual reproduction called conjugation. This is not mentioned in your textbook. Basically, when we talk about the sexual reproduction in bacteria, bacteria do not produce gametes and fusion of gametes, zygote formation, all of that drama will not happen. It's a very primitive type of sexual reproduction. If DNA transfer happens from one bacteria to another, that itself is considered as sexual reproduction. Okay, so one of the ways in which sexual reproduction can happen in bacteria is by conjugation. So some bacteria will have this pilus, this tube-like structure. Some bacteria will not have it. The bacteria that have it are called F+, the bacteria that don't have it is called F-. I'll just tell you very briefly. So what they do is, these F- bacteria will have a receptor for this. So this pilus will go and bind here, it will form like a continuous tube. And once that continuous tube will form, this will have a plasmid called the F plasmid. What color shade I use? Maybe green. Okay. So this plasmid, it makes an extra copy of this plasmid and that plasmid is transported to this. So exchange of DNA will happen. With the help of this plasmid, this F- also will become F+. It will also produce pilus and all of that. So basically that way this will help in a primitive type of sexual reproduction. And then you have structures that are called fimbriae. Fimbriae are very small bristle-like structures that sprout out of the surface of bacteria. You can think of them like the bristles in your toothbrush, like pointed structures very close to each other. Throughout the surface of the bacteria, you can find them. They're very small in length compared to flagella and pili, but their number is more. Okay, so what is their function? It helps bacteria attach to the host. If the bacterium is a parasite, it has to attach to the host. It could be a plant, an animal. Then this will help in attachment of the bacterium to the host's body surface, or if it's growing somewhere on rocks besides a river, like a stream is going there, there is wet place, it has to cling on to the rock, then it will help in attachment. When bacteria are growing in colonies, these bacteria attach to each other with the help of fimbriae. Basically, fimbriae are structures that help in attachment. This attachment could be to rocks, it could be to host tissues, or it could be in between bacteria within a colony. So here is an actual micrograph of a bacterial cell that contains fimbriae, flagella and pilus. You can see how there is this one long structure which is the flagellum. There is one thick tubular structure that you may or may not clearly see here. This is the pilus. Other than that, there are many bristle-like structures. Which color should we use? Pink. Why not? So many bristle-like structures that you can find here throughout the surface of bacteria. They are fimbriae. All of them arise from the surface of bacteria, but the functions of each of them is different. Okay. Now let's talk about the only organelle that you can find inside prokaryotic cells, which is the ribosomes. I mentioned how it is a universal organelle. You can find them in prokaryotes, you can find them in eukaryotes. Even though that is common to both prokaryotes and eukaryotes, still there are some differences between prokaryotic as well as eukaryotic ribosomes. So now just learn, let's just learn about prokaryotic ribosomes. Ribosomes are made up of two subunits. There's a large subunit and a small subunit. Ribosomes are structures or organelles in which proteins get synthesized. So ribosomes are called protein factories. You know, in eukaryotic cells, ribosomes can be associated with the nuclear membrane. They can be associated with the endoplasmic reticulum, making it rough in appearance, rough endoplasmic reticulum. Here, there is no nuclear membrane. There is no endoplasmic reticulum. What do they have instead? They have only one membrane, which is plasma membrane. So ribosomes are found associated with the plasma membrane in prokaryotic cells. Not all of them will be present associated with the plasma membrane. You can find some present in the cytoplasm as well. Now looking at their size, they are very, very, very tiny, super tiny. They measure in nanometers. They are wider, they are more wide than long. So the width is more compared to the length. The width is around, the length is around 15 nanometers and it's around 20 nanometers in width. Okay. Made of two subunits, the large subunit and the small subunit. That is mentioned as 50S and 30S. Now what does S stand here for? S here stands for Shalini, which is my name. Not really. It stands for sedimentation coefficient, which is measured in terms of a unit called Swedberg unit. Okay, so what is the Swedberg unit? It is something that measures indirectly the density of that particular organelle. Let's understand this. Whenever you're measuring weight, you use a weighing scale, kilogram, grams, like that. Whenever you're measuring length, you use meter, kilometer, millimeter, micron, nanometer, and these kind of units. Over here, we are measuring density. And how do we do that? We do that in an instrument called a centrifuge. A centrifuge is an instrument where you're spinning biological molecules and organelles and components at a very high speed. Okay, in an ultracentrifuge, you can spin these chemical components at speeds as high as 1 lakh rotations per minute, more than that rotations per minute. So when you're spinning something at very high speeds, the centrifugal force will act on those substances, right? Depending upon their density, they will settle down. Imagine you're taking like a handful of sand, you're mixing it in a glass that contains water and you're stirring it. If you allow it to settle for some time, you will notice that with time, the water will become clearer, the sand particles will settle at the bottom of the glass. If you notice, the larger particles are the first to settle. The smaller soil particles, the smaller sand particles will take a much longer time to settle. They'll be just floating around there and eventually with time they will settle down. Same thing will happen even over here. Whichever is denser will settle down quickly. Whichever one is not so dense will take a little longer and settle down. The time it takes to settle down is one factor. Where it settles down is another factor. Similar to how in your ruler, in your scale, you have this much is 1 centimeter, this much is 5 centimeters, this is probably 10 centimeters, this is 15 centimeter. In a centrifuge tube, there is a standardized unit or measuring sort of system where you can say if it settles at this point, it is 50S. If it settles at this point, it is 30S. So the more denser it is, it will settle down more at the bottom more quickly. The quicker it settles down, the more is its value. It's more dense. So this large subunit settles down over here, let's say, we call it 50S because it's standardized. We know that if something settles there, its value is 50S. And then if something is much lighter, it's smaller here, its density is also less. So this will be 30S. Now I want you to notice how this 50S and 30S, when they come together, they're not always together. They can exist as separate units, but when they have to do their job of synthesizing proteins, they will have to come together. Unless they come together, protein synthesis cannot happen. When they come together, what happens is the overall surface area will reduce, right? That is exposed, will reduce. And also, you may have learnt in physics how when objects containing two different masses come together, they are interlocked together, their overall density will reduce. It's not like 50 plus 30, the density right now will be 70, 80s. It will not add up to 80, it will become 70. Here we are not doing high school arithmetic math, right? So here, this is not about addition. It is about what is the density, at what point is it settling. Obviously, when two objects are coming together, its overall density will get reduced. As a result, they will not settle down at 80. They will not be 80s. They will settle down at 70s. Okay. So this is not additive. Be very sure of that. 50 plus 30 is not 80. 50 plus 30 is 70s. Here we are not counting 8 oranges plus 30 oranges, 38 oranges. Here we are looking at the sedimentation coefficient, where it is settling, how long it is taking. That is what is important here. Okay. So that's about the ribosomes. Now, I already mentioned that ribosomes are where proteins get synthesized. Sometimes in prokaryotic cells, what happens is when proteins get synthesized, it's a complicated process. You will learn about it in another chapter. For now, what is required, I'll tell you. The information for protein synthesis comes from a molecule called mRNA. mRNA is carrying that message. The message is read inside the ribosome. While reading the message, simultaneously polypeptide or protein synthesis happens. It is possible that sometimes one mRNA carrying information, one single molecule, one single strand of mRNA carrying information can be bound to several ribosomes. As you can see here, this is one mRNA. This is bound to 1, 2, 3, 4, 5, 6 ribosomes. So it's like they're all moving like this. This ribosome first attached, it read the message, see such a long chain of protein it has formed. The next one started here, it read the message, it reached there, and it's formed such a long chain. You can see how they are all forming chains. They are reading the message from here to here. So whenever to a single strand of mRNA, many ribosomes are attached, you call such a complex a polysome or a polyribosome. Poly means many. Polyribosome means many ribosomes. Where are these many ribosomes? These ribosomes are attached to a single mRNA molecule. They form, it's like one mRNA is there, to that many ribosomes are attached. So you call that structure polyribosome or a polysome. Okay. Now, we've almost reached the end of the chapter. We'll talk about inclusion bodies. You know how in eukaryotic cells also, certain cells are specialized to store reserve food. Even in our body, for 2 or 3 days, if we don't eat, if we fast, or if you're starving, you didn't find food, you're stuck in an island, we will not die. Okay, because in our body, whenever we've eaten excess food, it would have stored, gotten converted into glycogen or fat and stored in our body. We can use that reserve food. Likewise, even plants will store reserve food in the form of starch. When there is not sufficient sunlight, when there is not sufficient water, or maybe carbon dioxide is not enough, it will not photosynthesize very well. Whatever is stored, it will make use of it and continue to survive. Likewise, bacteria also need some things to survive. When they are not capable of finding food, when they are not able to synthesize food if it's photosynthetic, they should have some reserve food that it can use later. Not necessarily food, sometimes they store phosphates as reserve. Okay, so whatever is required for their functioning, they store it. That is stored in the form of inclusion bodies. Inclusion bodies again are not bound by any membrane. They are granular in nature. You can find them freely present in the cytoplasm of prokaryotic cells. Okay, so they are called inclusion bodies and they are not bound by any membrane. You have examples of 3 different types of inclusion bodies in prokaryotic cells like bacteria. Some of them are called phosphate granules. They store, obviously, as the name itself indicates, phosphates. So whenever ATP synthesis, adenosine triphosphate, phosphate synthesis, ATP synthesis or anything has to happen, that they can use that phosphate from this reserve. Some cyanobacteria store reserve food in the form of cyanophycean granules. That is mostly stored as protein. Sometimes it could be stored as starch as well. Okay, so that is found only in cyanobacteria. And then glycogen granules, they store glycogen and that is a carbon source. You can find that in most bacteria. That's about inclusion bodies. Now moving on to the last part of prokaryotic cells, gas vacuoles. These are super cool structures that you can find in photosynthetic aquatic bacteria. Okay, so there are bacteria called green sulfur bacteria and purple sulfur bacteria. Also, there are blue-green algae, which is nothing but cyanobacteria, all of these are photosynthetic. Okay. Not only are they photosynthetic, they are also aquatic. They are found in water. You know, sometimes for these photosynthetic bacteria to perform photosynthesis well, sunlight is essential. They are required to float on the surface of water so that they can get maximum sunlight. Similar to how we have these life jackets, that will help us float on the surface of water and make sure that we don't sink. These also have life jackets that they don't wear outside, but that is an inbuilt jacket that it is produced inside. Whenever they are not receiving sufficient sunlight, the cell itself, certain genes will get activated and it will start synthesizing vacuoles called gas vacuoles. Remember that vacuoles are also present in plant cells, large central vacuole that is bound by a membrane. These vacuoles are not bound by a membrane, they are bound by a rigid protein sort of structure inside which gas is present, literally air is present. Okay. So when this inbuilt life jacket is produced inside the bacterial cell, it will allow the bacteria to float on the surface of the water body. It could be a pond, river, wherever it's growing. In that aquatic environment, it can float there. And it's also possible that sometimes the sunlight can become too harsh for the bacteria, in which case the bacterial pigments could get damaged. When the sunlight is too harsh, then what they do is they will pop that gas vacuole, poof, it is gone. The gases are released outside and then it will kind of, lose that buoyancy and settle down. Okay, it will sink down a little bit. So the presence of these gas vacuoles will allow these photosynthetic aquatic bacteria to float at a level that is convenient for their growth and photosynthesis. Okay, so this is so clever, right? We think that bacteria are very primitive. They don't have a brain. They're not even multicellular. But think about it. Despite being so simple in structure, they are so clever, and they've come up with so many ways to ensure and maximize their reproductive fitness, to make sure that they continue to exist and reproduce, and their species continue to exist for several generations, right? So that's about it. Now we have to start learning about eukaryotic cells. Let's get started with that. Now let's take a look at some examples of eukaryotic cells. Other than bacteria, most other organisms are eukaryotic. For example, your protozoans like amoeba and paramecium, they are eukaryotic. Slime molds are eukaryotic. Dinoflagellates and diatoms are eukaryotic. Other than that, all fungi, all plants, all animals are eukaryotic, meaning they have cells. Within the cells, there is a well-defined nuclear membrane, and within the nuclear membrane, you can find DNA. So basically, the genetic material is enclosed by a nuclear membrane, or you can call it the nuclear envelope. Inside the nucleus, DNA or the genetic material exists, and sometimes, depending upon which phase the cell is in, which phase of the cell cycle the cell is in, it can even form chromosomes. Now, the only difference between prokaryotic cells and eukaryotic cells is not about the nuclear membrane. Similar to how prokaryotes lack a nuclear membrane, they also lack other membrane-bound organelles like our endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, chloroplasts. None of those organelles exist in the prokaryotic cells. I've been saying since the beginning that prokaryotic cells are kind of primitive cells and eukaryotic cells are more advanced cells. So what is the difference between these two? One of them does not have any membrane-bound organelles and the other one has a lot of membrane-bound organelles. It's more crowded, right? How does having more organelles equate to being more advanced? Unless there is an advantage of having those organelles, you don't call it advanced, right? What do you think is the advantage of having membrane-bound organelles like the ones that I just mentioned? I'll give you a very simple analogy to make it easy for you to comprehend this. Let's take one house for example. In this one house, there are no cross walls at all. There is just one outer boundary wall and a roof, but you don't have walls in between demarcating different rooms. Within this single space without any walls. All of the activities inside the house are going on. Food is stored in this storeroom. Okay, there is no partition though. Food is stored there. The kitchen is here. Let's say the bathroom is here. And let's just say the living room here. The living area is here, not living room. The living area is here. So imagine what the state of the house would be had the house been this way with no partition at all. Absolutely no partition anywhere. If you're taking a shower in the bath area, the water will leak out and flow into the kitchen, which is so unhygienic, right? So the food that is stored right next to the bathroom or the bath area, you would have stored some grains there or some vegetables there, let's say. If it is not inside an enclosed area, if it's not protected, then the dirty water from the bathroom or the bath area could reach this region as well. If you had like a puja area here, the dirty water could enter the puja area as well, right? I'm just giving you a simple example to make you understand how complicated the biological processes can become if there is no compartmentalization. Imagine one cell, let's say a plant cell. If this is like one plant cell, in a plant cell, photosynthesis has to happen. Cellular respiration also needs to happen. In photosynthesis, glucose is synthesized. In cellular respiration, glucose is broken down. If the enzymes required for synthesis of glucose is located in the same space where enzymes for glucose breakdown are located, how efficient can either of the processes be? It's going to be super inefficient, right? By the time glucose gets synthesized, there could be some enzyme that acts upon the intermediates and break it down, right? So that can happen. So becoming eukaryotic cells or evolving into eukaryotic cells, one thing has happened which is amazing is compartmentalization of the cytoplasm. How exactly is the compartmentalization happening? In our houses, we build walls to form compartments, different areas. There are different activities that take place. Inside a cell, what kind of wall can you build? You don't build brick walls inside of a cell. Inside of a cell, there are membranes. The membranes itself will kind of form compartments within the cytoplasm. Okay? So, because of the presence of membrane-bound organelles, the cytoplasm becomes compartmentalized, meaning it will form two different compartments. The membrane-bound organelle chloroplast will house all the enzymes required for photosynthesis. The membrane-bound component mitochondrion will house all of the enzymes required for cellular respiration. So they are physically separated. It's happening in two different compartments. So it's going to be super efficient, right? So compartmentalization has made sure that the biological phenomena, processes that take place inside the cell happen very efficiently. Now, apart from compartmentalization, another advancement that you can see in eukaryotic cells over prokaryotic cells is the fact that eukaryotic cells have more complex locomotory structures as well as cytoskeletal structures. Locomotory structure is very simple to understand, structures that will help organisms move. It will contribute to motility. Flagella is present even in bacteria. Here also flagella is one of the locomotory structures, but the way it functions, the structure of it is very, very different. When you talk about cytoskeletal elements as well, cytoskeletal elements are those proteins that form the skeletal system of a cell. Okay, similar to how we have a skeleton, each cell has a skeletal system. But unlike our skeletal system, which is made up of bones and cartilage, the skeletal system of a cell is made up of proteins. Okay, so just like how our skeletal system will provide us structure and shape, the cell is maintained in that particular shape because of the presence of these cytoskeletal elements. Of course, they exist even in prokaryotic cells, but in eukaryotic cells, they are far more elaborate. Okay, that way also eukaryotic cells are slightly more advanced than prokaryotic cells. Now, other thing that I mentioned was that here in eukaryotes, the genetic material is organized into chromosomes. Now, next, you know, eukaryotic cells include plant cells and animal cells. I already mentioned that. Plant cells and animal cells differences you may have learned in your lower grades as well. Let's quickly recall what are some of the differences between plant cells and animal cells. Here is an illustration from NCERT of a plant cell. This is a diagram of an animal cell. Plant cells have a cell wall which animal cells don't have, right? So, cell wall is a structure that exists in plant cells alone. This large central vacuole exists only in plant cells. You don't find them in animal cells. Is there anything that you find in animal cells which is absent in plant cells? Of course. In the animal cells, you find centrioles or centrosomes, which you don't find in plant cells. The plant cells also have organelles called plastids. A very popular type of plastid is chloroplast. It helps in the process of photosynthesis. That is also an organelle that you find only in plant cells and not in animal cells. Of course, there are many different types of eukaryotic cells, fungi, plant cells, animal cells. All of them are eukaryotic because in all of them, surrounding the genetic material DNA, there is a well-defined nuclear membrane. That does not mean they all are similar, they all are identical. Even though they are eukaryotic, there will be certain fundamental differences between plant cells and animal cells. Okay, now let's talk about the cell wall. Cell wall is a structure that you don't find in all eukaryotic cells. You can find them in plant cells, you can find them in fungal cells, but you don't find them in animal cells. Okay, we'll start with the cell wall and we'll move inside. Similar to how we did with prokaryotic cells, we started from outside and moved inside. We're going to do the exact same thing while understanding eukaryotic cells as well. So the cell wall in eukaryotes is a non-living kind of rigid structure which forms the outermost covering of the plasma membrane. You can find them in fungi, you can also find them in plants. What is the function of the cell wall? Like any wall, the cell wall provides a proper shape to the cell. It offers protection against any damage or any pathogens. It helps in cell-to-cell interaction. There are structures like certain junctions present between neighboring cells which pass through the cell wall. Like in plant cells, there are plasmodesmatal connections about which we'll learn, through which communication between neighboring cells can happen. In order for a multicellular organism to exist and survive, cells, many cells in our body will have to communicate with each other. They don't talk in English or Hindi or Malayalam and communicate with each other. They communicate through chemicals. So there should be some provision for chemicals from one cell to move to the other cell via the cytoplasm or any other cellular junction. So that is possible because of the cell wall. That can be made possible because of the cell wall. And this acts like a barrier to undesirable macromolecules. The cell wall is made up of mostly cellulose in plants. That also is like a mesh-like structure. If the molecules are very small solutes and all of that, it will freely allow it to pass through it. But if the size of the molecule is big, like very large proteins or very large polysaccharides try to enter or leave the cell, it will not allow that. Okay, so it acts like a barrier. Different groups of organisms have different components in its cell wall. In many groups of algae, you can find, of course, the presence of cellulose like all plant cells. But along with that, you can also find presence of galactans, mannans, minerals, and substances like calcium carbonate. Many plants, like higher plants, pteridophytes, gymnosperms, angiosperms, in those cells, you can find cellulose, hemicellulose, pectins, and certain proteins as well. In fungi, the cell wall is made up of chitin. In this class, in this chapter, we are only going to focus on how is the cell wall in plants. We will not learn about the structure of fungal cell wall or other lower groups of plants, algal cell wall. In higher plants like in angiosperms, how does a cell wall form? What are the different layers? We learn about that. Now, yeah. So, when a brand new cell wall will form in plant cells, let's take one plant cell for example. This is one big plant cell. This plant cell is too big. Now it has decided to undergo cell division. So this plant cell's nucleus has already divided. Karyokinesis is over. Now the two cells have to divide. The cytoplasm has to divide by a process called cytokinesis. So the cell wall has to form between these two cells. Remember, plant cells don't just have the cell wall, they also have what is called the cell membrane. So inside the cell wall, the cell membrane or the plasma membrane also will be present. Okay. Now, once the nucleus of the plant cells have divided, have separated, first, the cytoskeletal elements present in this particular plant cell will form a sort of framework in between these two nuclei. Oh my god, that became too big. Let me reduce its size. Okay. So this is formed from the cytoskeletal elements. Let's just call this scaffold. This kind of gives like a framework for the new cell wall to form. The proteins start aligning there and they say like this is the point where the new cell wall has to form. In the cell wall, there are many layers present. The first layer that forms when two cells divide is called middle lamella. This middle lamella is made up of a substance called calcium pectate. Pectin is a polysaccharide. Pectin is produced inside Golgi apparatus. So inside this cell, there will be Golgi apparatus, right? Let me represent that using yellow color itself. Notice and understand that the structure of the plasma membrane is very similar to the structure of the Golgi membrane. This is not the most proper way of representing a Golgi apparatus or a Golgi body, but for our understanding, I've just drawn a sphere over here. Inside this Golgi, you can find pectin produced. Okay, so Golgi will produce pectin. It will release that in the form of this vesicle. Now this happens on both sides. Now this membrane moves towards the scaffold. This will start moving here. Once it moves there, it kind of fuses with the existing plasma membrane. Okay, imagine that it went very close here and then the membrane fused with the already existing plasma membrane and then it released all of the pectin over here. Okay, so outside I copy slide. 1 minute, not even a minute. Okay, we're done. Yeah. So the pectin gets deposited near the scaffold. As a result, what happens is, the first layer forms here. The middle lamella forms here. Okay. So this is middle lamella. On either sides, you can find right next to the scaffold, middle lamella will form. And what is this middle lamella made up of? It is made up of calcium pectate. First formed layer, outermost layer. How is it formed? It's formed from the pectin that was produced in the Golgi, transported by the Golgi vesicle and released near the scaffold. So this is composed of calcium pectate. Now as the cell begins to grow, newer and newer cell wall materials will get deposited. We've learned how cell wall is mostly composed of cellulose in our lower grades. Even today I mentioned how plant cells are mostly made up of cellulose, but cellulose is nowhere in the picture right now. What is cellulose? Cellulose is basically a polysaccharide made up of repeating units of glucose. The enzyme that is responsible for the synthesis of cellulose is located in the plasma membrane. This is the plasma membrane. I told you that they fused, right? So, over here in this membrane, there are enzymes called cellulose synthase on both sides. Now, what the cellulose synthase does is, it will take the glucose from the cytoplasm. It will start picking up one glucose after the other and it will join it to make a long chain of cellulose and push it outside. Okay. So, if anything newly is getting formed, it has to form towards the living side of the cell itself, right? So, the newer cell wall that will form inside of middle lamella, existing middle lamella. So, from here, the cellulose will start getting pushed outside and cellulose will form in between the middle lamella and the plasma membrane. Okay. So, let me represent that here. This is the cellulosic cell wall. Okay. So, this is called the primary cell wall. This is made up of cellulose, of course, which is formed at the plasma membrane. It's not transported from inside the cell, it gets formed at the plasma membrane. Along with that, there will be small amounts of hemicellulose and some amount of pectin as well. Cellulose and hemicellulose are kind of rigid structures, but pectin is not like that. You know, pectin is used in jams and spreads also because of its glue kind of nature. It is gum-like, so it spreads very easily. In your jams that you use, Kizan jam between your bread slices, that also they use pectin because it's gluey. Okay. So that is kind of like a soft glue that holds the two neighboring cells together immediately after the cells have divided. But after that, more rigid solid structures will start forming towards the cytoplasmic side. So you have the middle lamella, right next to it you have the primary wall, which is made up of cellulose, hemicellulose and pectin. If the cell continues to grow, there will be proteins or enzymes called expansins that will make sure that the cell wall will get extended. The cell wall, the cellulose will start getting incorporated into the primary wall and the cell will increase in size. Okay. Now in some cells only, inside the primary wall, a secondary wall also can form. This is not present in all cells. This is present only in a few cells such as sclerenchyma cells or xylem elements like trachea or tracheids. You can find them. That is called secondary wall. The secondary wall is not always present. It may be present in some cases. What it is composed of varies from cell to cell. It could be composed of substances like lignin. It could be composed of substances like suberin. Depending upon the cell type, the secondary cell wall composition varies. So this is the ultimate cell wall structure. Outermost, you will have the middle lamella made up of pectin. Inside the middle lamella, you will have primary wall made up of cellulose, hemicellulose and pectin. Inside of that, you may have a secondary cell wall which is made up of either suberin or lignin depending upon the cell type. Inside that, you have the cell membrane. This is from outside to inside. These three layers are present in the wall. Third layer may or may not be present, but the other two layers will for sure be present. Outermost is the middle lamella. Inside that, you have primary wall. Inside that, you have secondary wall. Inside that, you have plasma membrane. Inside the plasma membrane, of course, you will have the cytoplasm. Now, how does this lignin or suberin get transported to the membrane, towards the membrane for it to get deposited as the secondary wall? The same way pectin gets transported by means of vesicles only. Pectin, suberin, lignin, all of them are sent towards the cell wall by means of vesicles. Cellulose gets synthesized at the membrane. Okay, that process takes place at the membrane. Others, they are synthesized, the materials are synthesized by the Golgi, they are transported by Golgi vesicles, and then later they reach the cell wall. Okay, now let's talk about cell membrane. Cell membrane, we are studying under eukaryotic cells, but whatever structure you will study for cell membrane over here, the same thing will apply even to prokaryotic plasma membranes. The same thing will apply to all of the membranes that you find inside the cell. The nuclear membrane, the ER membrane, the Golgi membrane, lysosomal membrane, vacuole membrane, mitochondrial membrane, plastid membrane, all of them have a similar structure. Whenever I say membrane, assume that I am talking about this structure. Okay. Now, how did we get to know about the structure of the plasma membrane? Initially, electron microscopy was not so advanced when they started to study the structure of plasma membrane in living organisms. We got to know about that only in the 1950s. But prior to that, before 1950s, biochemical studies had been made on cells. People extensively studied erythrocytes or red blood cells. And when they studied that biochemically, they tried to determine what all chemicals are the cell membranes made up of. They figured out the cell membranes are mostly made up of lipids and proteins. Okay. So they said that cell membrane is made up of two substances, lipids and proteins. First, let's talk about lipids. They say that the lipid that they found in the plasma membrane was mostly phospholipids. These phospholipids have a typical structure. They have a phosphate head and two hydrocarbon tails. Now, this phosphate head likes water, is hydrophilic. The hydrocarbon tails do not like water one bit. They are hydrophobic. What we know is that inside of our cells, if this is the plasma membrane, inside of our cells, there is the cytoplasm, and the cytoplasm is mostly made up of water. Outside the plasma membrane, there could be interstitial fluid or extracellular fluid, which also will contain some amount of water. Now, if these phospholipids are the major component of the plasma membrane, if the phospholipid is located like this, then there is a problem, because for the head it is okay. Head is facing towards a water containing region, it's fine, it does not bother about it. But the hydrocarbon tails that don't like water are facing cytoplasm which contains water. They cannot stay like that, they hate water. If it is the other way, if it's like this, then this part again will be okay with it, it does not mind, but this part does not like it. What if it's like this? Then again, the part where the membrane, the phosphate head is there would not mind. The part where hydrocarbon tails are there, they will not be okay being around water. So what they figured was, phospholipids are there, there's water on either sides of the membrane. It's not possible for phospholipids to be arranged as a single layer. So it's possible that the phospholipids are arranged in the form of two layers such that both the places where water is there, okay, so there's water here, there's water here, wherever water is being faced, over there the phosphate heads are there and the hydrocarbon tails are facing inwards away from water. So they figured that these phospholipids existed in the form of two layers and that is called a phospholipid bilayer. Apart from phospholipids, within the cell membrane, they also found cholesterol present. Cholesterol is also a lipid. Okay. So this cholesterol would be present between here and there, in between the phospholipids, and that gives stability to the membrane. When I'm saying that the cell membrane or the plasma membrane is made up of lipids, I mean to say that they have oil-like consistency. In fact, researchers even describe the plasma membrane to have consistency like olive oil. Okay, so they are kind of fluid. They are not like solid, hard structures. They are very fluid, almost quasi-fluid we say, semi-fluid of sorts. They have oil-like consistency, similar to how when you keep coconut oil at home during winters, it tends to solidify, and then during summers, it liquefies. Exactly that can happen with our cell membranes as well. In order to make sure that our cells do not become like overly rigid during winter when the temperature is low, and it becomes and to make sure that it does not become overly fluid and leaky during summer, these cholesterol molecules will help. Cholesterol is very good. It plays a very important role inside of our plasma membrane. But if the same cholesterol is present inside your bloodstream, then it is a problem. In a membrane, they are meant to be there. They are doing their job very well. Okay, cholesterol is not bad if it's present in your plasma membranes or any membrane for that matter. It makes sure that the consistency of the lipid, of the phospholipid bilayer is maintained even at all temperatures, too high, too low. It makes sure that it is in a normal consistency. Okay, so that's how it, provides stability to the plasma membrane. So here is the diagram of a phospholipid bilayer. You can see how over here there are phosphate heads and hydrocarbon tails. This is the outer surface of the plasma membrane. This is the inner surface towards the cytoplasm. Okay, so you can see like it is a continuous layer present. I drew it in 2D and showed it to you, but this is more like a 3D diagram. I want you to imagine it in the form of a sphere. If the cell is like a sphere, throughout the inner surface of the cell you have phosphate heads, throughout the outer surface of the cell you have phosphate heads, in between them you will have these hydrocarbon tails. Okay, so you can notice in this diagram there is these cholesterol molecules also shown. Okay, now that's about the lipids. Lipids are mostly phospholipids. They are arranged in the form of a bilayer. Polar head or hydrophilic head faces the outer sides, hydrophobic tails face the inner side. This makes sure that the non-polar tail of the saturated hydrocarbons is protected from the aqueous environment, which is water. In addition to phospholipids, the membrane also contains cholesterol, like I already told you. Now talking about proteins. Depending upon how easily proteins can be extracted from the membrane, you can categorize them into two types. One is called peripheral protein, the other one is called intrinsic or integral protein. So how are these peripheral proteins? This is a protein that is spanning the entire membrane here, right? It's just itself inside this phospholipid bilayer. It's just projected inside the phospholipid bilayer throughout, right? If it's completely penetrated the phospholipid bilayer or at least partially penetrated the phospholipid bilayer, as you can see here, you call them integral proteins. Okay, so it's mentioned here. See, this is an integral protein. There could be proteins that are present just towards the surface like that. Such proteins are called extrinsic proteins or Peripheral proteins. Of course, if it is not present throughout the membrane, spanning the membrane, it's easier to isolate that. It's easier to extract that from the membrane. So this is easier to extract compared to this. This is called peripheral protein. The classification is based on how easily you can extract that from the membrane. Okay. Now, later, further biochemical investigation helped us find out not only does the membrane have lipids and proteins, it also consists of carbohydrates. Now, these carbohydrates may either be associated with proteins or they may be associated with the lipids. For example, if you can look here, there are these Y-shaped structures that you see, right? These are nothing but sugars, okay, oligosaccharides, very, very short chains of monosaccharides. Here you can see there is a protein, integral protein. To this protein, this sugar is attached, right? Here you are seeing a sea of lipids. To this lipid, this sugar is attached. So carbohydrates usually towards the outer side of the membrane. You don't notice that inside, right? Towards the outer side of the membrane, you can see how they are either associated with proteins or they are associated with lipids. If these carbohydrates are associated with the protein, then the protein is called a glycoprotein. If the carbohydrate is associated with the lipid, then the lipid is called a glycolipid. Okay. The percentage of proteins and carbohydrates, proteins and lipids in different cells varies. In some cells, there's more lipid than proteins. In some cells, there's more protein than lipids. Since RBCs are the most extensively studied type of cells with respect to the structure of plasma membrane, in RBCs, you can notice that protein content is more than lipid content. If proteins are around 50% within the plasma membrane, then the lipids are, 52%, then the lipids are just about 40%. Okay, that varies from cell type to cell type. Now, let's talk about why the structure of the plasma membrane is called a fluid mosaic model. This is a model that was proposed by Singer and Nicholson in the 1970s, in 1972. And this kind of explains the structure of the plasma membrane. According to them, the nature of the plasma membrane is quasi-fluid. Okay, that means it's semi-fluid. It's not a rigid structure as you think of it. The membrane is not like a thin cotton cloth. It's not like a thin paper. It is actually like olive oil in consistency. If you find like a super tiny, very thin needle, if you prick it, it will go inside the plasma membrane. That's how fluid the plasma membrane is. It's not a solid structure. It's semi-fluid. Okay. So, because how is it semi-fluid? It's fluid because within the membrane, the phospholipids can move. It's not like this phospholipid that I've drawn right here is fixed in position. It's going to stay here forever. It can move within this membrane. So can proteins move within the membrane. Because they are moving, they are fluid, the consistency of the plasma membrane is oil-like. Okay. Yeah. The fact that the plasma membrane is fluid-like explains many functions of the cell. If a cell has to grow, if it has to increase in size, if the cell membrane is a very rigid structure like one solid box, then how can it grow bigger? Unless it is fluid, it cannot grow bigger, right? If it has like a very rigid structure. So it explains that. Secondly, formation of intercellular junctions. The junctions between two cells, be it gap junctions, tight junctions, or even plasmodesmata for that matter, that can happen because of the fluid nature of the plasma membrane, secretion by means of exocytosis, endocytosis, taking up substance from outside the cell into the cell because of engulfing substances because of its fluid nature, secretion, cell division. For animal cells to divide, the cell membrane has to pinch itself. It has to form a furrow. That is not possible if it's like a solid box. All of these processes can be explained because of the fluid nature of the plasma membrane. Now, the plasma membrane performs many functions. One of the most important functions of the plasma membrane is that it allows transport of substances to happen from the inside of the cell to the outside, from the outside to the inside as well. So now, the cell membrane is said to be selectively permeable. It does not allow anything and everything to pass through it. It carefully selects what substances can move into the cell, what can leave the cell based on its size, based on its polarity, based on a lot of conditions inside the cell. That's why it's said to be selectively permeable. Now, across the plasma membrane, movement of molecules can happen by means of various processes. It can happen either passively or it can happen actively as well. Let's see what are the various ways in which passive transport can happen, then we learn about active transport. Okay, so there are 4 types that you need to learn about. The first 3 are passive transport. The last one is active transport. Okay, these ones are passive. The last one is active. In passive transport, you have 3 types. There's simple diffusion, facilitated diffusion, and osmosis. What happens in simple diffusion? Let's try and understand. For simple diffusion to take place, a membrane is not essential, but even in the presence of a membrane, it can happen. I'll show the membranes here. Okay, so let's think of this structure that I'm drawing here as the plasma membrane. If movement of substances happens from a region where its concentration is more, okay, its concentration is more here, its concentration is less here. So if movement of substances happen in this direction from where it is already high to where it is low, across the plasma membrane itself, across the phospholipid bilayer itself, then we call it simple diffusion. What is facilitated diffusion? Facilitated diffusion is when there's more substance on one side. Okay, so there's more on one side for sure. There's more on this side. Fewer molecules on this side. The concentration of the substance is more on this side and less on this side. Let's write low. Movement will happen from an area where concentration is more to a to where the concentration is less. But if these molecules are polar, they won't be able to pass through the phospholipid bilayer here. They are hydrophilic molecules, they are polar molecules, charged molecules. So they will not be able to pass through the phospholipid bilayer themselves. In order to help these polar molecules, the plasma membrane will have certain proteins present. These proteins are integral proteins and they will form channels through which transport will happen. Since the diffusion here is facilitated by these proteins present in the membrane, you call this facilitated diffusion. What's common to both is that transport of substances is happening from a region where the concentration of molecules is high to a region where the concentration is low. Here it is happening via the phospholipid bilayer itself. Over here that is not possible because this substance is polar. So it will happen by means of proteins present in the membrane, integral proteins present in the membrane, and since it is facilitated by these proteins, it is called facilitated diffusion. When it comes to osmosis, the third type of passive transport, it's about molecules of water. From a region where water molecules are high to a region where water molecules are low. If water is being transported across the semi-permeable membrane or the plasma membrane, then you call it osmosis. Osmosis is specifically the movement of water across this membrane. So in all these cases, what you noticed was the movement of substances is happening from a region where the concentration is high to a region where the concentration is low. Of course, there is a gradient here. There is a difference in concentration. From where it is more to where it is less, it is moving. It is happening along the concentration gradient. Since it's happening along the concentration gradient, it is a spontaneous process, does not require ATP. As opposed to this, what happens in active transport is, if there is less molecules on one side and more molecules on the other side, okay, so gradient exists here, there is a difference in concentration here. On one side the concentration is low, on the other side the concentration is high. Even though the concentration here is already high, these substances will be moved towards this part where the concentration is already high. So here the movement of substances is happening against the gradient. Since it is happening against the gradient, that looks so bad. Since it's happening against the gradient, it requires ATP. It requires energy in the form of ATP. Now, think about how water falls from the fourth floor to the ground. You don't need to put too much energy. It spontaneously, just because of gravity, falls down. If you were to pump groundwater up to the fourth floor, you need motors, you need electricity, you need energy, right? Same thing here. If you have to move substances from a region where it's already low to a region where it's already high, it is not a spontaneous process. It requires energy and it requires proteins present in the membrane that are called pumps. So these pumps make use of ATP, and using the energy from ATP, they will pump molecules against their concentration gradient. One example for this is the sodium-potassium pump. You know, more about the sodium-potassium pump and how it works, you will learn in neural control and coordination. Basically, for every 3 sodium ions pumped in, 2 potassium ions will be pumped outside by making use of 1 ATP. Okay, so that is how active transport is. Passive transport always happens along the gradient, no need of ATP, but there it happens against the gradient. You can see how here also proteins are used, here also proteins are used, but the difference is these are just facilitating the transport of molecules from here to here. They are providing an environment where molecules can easily move. This is not consuming any energy in the form of ATP, whereas here ATP is utilized for molecules to get pumped from one side of the membrane to the other side. Okay, now that's about the plasma membrane. Now let's talk about the organelles that we can find in eukaryotic cells. It's very interesting. You have very many organelles that you can find in eukaryotic cells. There's mitochondria, there's chloroplast, peroxisomes, vacuoles, Golgi apparatus, lysosomes, all of them. Okay, but some of these organelles functions happen in a coordinated manner. All such organelles are grouped together as the endomembrane system. Endo means what? Endo means inside. Inside the cell, if this is the cell, inside the cell, there are many membrane bound organelles. Okay, so there is chloroplast, there is nucleus, there is endoplasmic reticulum, there's Golgi apparatus. Okay, so there is vacuole, and there is, let's say, lysosome. So many different types of organelles are found. Some of these organelles that you find in the cell, some of their functions are coordinated. Think about it. When you're constructing a house, when you're planning your house's layout, you do not want your storeroom to be in one corner and your kitchen to be in another corner and your dining area to be in one other corner. You want to make sure that the storeroom and the kitchen and the dining area are very close to each other. Why? Because when you're cooking, you might want to pick up some grocery from your storeroom. So if it's closer, it makes the job easier. If the dining area is situated very close to the kitchen, then once the food is prepared, it's easier to serve. It's not like your dining area will ever be inside a bedroom, right? Inside the bedroom, dining area will not be there. Inside the bathroom, storeroom will not be there. There is a reason. It is planned. Houses are planned that way because it makes work easier, right? Dining area is always close to the living area. Even while you're enjoying your meals, you can watch TV in the living room. So that is how usually houses are designed, right? Very similarly, even in organelles, you have different organelles performing their own functions. But the functions of some organelles are such that they will have to communicate with each other by means of chemicals, by means of vesicles. So all of those organelles whose functions are kind of coordinated, they depend on each other. Such organelles form a system inside the cell that we call as the endomembrane system. The output of one organelle could be the input of another organelle. That's how it works. When we understand these different organelles, you'll get a clearer picture about it. So now just understand these four organelles that I'm about to mention, they all function as a system and their roles are coordinated. Which are the four organelles? We have the endoplasmic reticulum, we have the Golgi apparatus, we have lysosomes, and we have vacuoles. Okay? So in this endomembrane system, mitochondria, chloroplast, peroxisomes, these organelles are not included. Okay? Now we'll have to learn about the role of each organelle. What is its structure like? What are its functions like? Okay, so we'll get started with endoplasmic reticulum. Endoplasmic, endo means inside, plasm refers to the cytoplasm, reticulum means network. Okay, so endo here is inside. Endo side. Plasmic refers to the cytoplasm. Reticulum means network. Inside a cell, you have the cytoplasm here. Within the cytoplasm, the endoplasmic reticulum forms a network. You can see how it is arranged in the form of sheet-like structure. Throughout the cytoplasm, it can be spread sometimes. It spans from the nuclear membrane till the plasma membrane sometimes. So, it forms a network sort of a structure inside the cytoplasm. Now, the presence of the membrane of the endoplasmic reticulum divides the space inside the cell, the intracellular space, into two distinct compartments. How many compartments? Into two compartments. What are they? Inside the endoplasmic reticulum, it's not empty. Okay, there is an aqueous solution similar to the cytoplasm inside the endoplasmic reticulum. It's also mostly water, certain enzymes, proteins, ions will be present there. So the presence of this membrane has divided this intracellular space into two compartments. One compartment is what I'm highlighting right here in green. It's not very visible, so let me use a pink color here. Pink is also not very clear. Yellow may be the right choice. Okay, so inside the membrane, inside this membrane, you can find this space inside, right? This space that you find towards the inner side of the membrane, this is called lumen. It's not empty space again. Within this space, you will find water, minerals, ions dissolved, certain enzymes and all of that. So this part of the intracellular space is called the luminal compartment. If you look at the other side of the membrane, which right now I will show in pink color, so this side of the membrane, everything outside the membrane here towards this side. So this is outside the lumen. You call this the extraluminal compartment. It is called the extraluminal compartment. Inside the cytoplasm is extraluminal. Within the lumen of the ER, that is the luminal compartment. Now, ER is of two types. The first type is called rough endoplasmic reticulum. The second type is called smooth endoplasmic reticulum. This rough endoplasmic reticulum arises from the nuclear membrane, outer nuclear membrane. The smooth endoplasmic reticulum is not directly connected to the nuclear membrane. It is arising from the rough endoplasmic reticulum. In this diagram, you can see very clearly. I will show in red color. This is the nuclear membrane. Continuous with the nuclear membrane, you can find the membrane of rough endoplasmic reticulum. Okay. But on this side, you have smooth endoplasmic reticulum. Smooth endoplasmic reticulum has no connection with the nuclear membrane. They are directly arising from the rough endoplasmic reticulum. What is the reason why rough endoplasmic reticulum is called rough endoplasmic reticulum? The reason is simply that when you look under the microscope, this has a rough appearance, that has a smooth appearance. Why does this have a rough appearance? Because on top of this rough endoplasmic reticulum, you can see the presence of ribosomes. Many ribosomes are present on the membrane of these rough endoplasmic reticulum. If you look at this, you don't find the presence of ribosomes, so that is smooth in appearance. Now, what is the function of rough endoplasmic reticulum inside a cell? It helps in protein synthesis. Again, I repeat, I'm saying that this helps in protein synthesis. It does not produce proteins itself. Protein formation can only happen in the ribosomes. When I discussed the organelles and the functions of prokaryotic cells, I mentioned how ribosomes are protein factories, right? So, protein synthesis can happen only in ribosomes. These are providing surface area for ribosomes to attach and thereby helping in the process of protein synthesis. They are involved in protein synthesis, but they are not directly taking part or contributing to protein synthesis. Okay. Now, coming to smooth endoplasmic reticulum, these are involved in the synthesis of lipids. In whichever cells of our body, lipids are constantly getting synthesized. Steroidal hormones are getting synthesized, like your testosterone, estrogen, progesterone. All of these are steroidal hormones, right? So they are also lipid-like. So wherever such hormones are getting synthesized in such cells, you can find these smooth endoplasmic reticulum to be spread out across the cytoplasm in large quantities. Okay, so these continue from the smooth endoplasmic reticulum and are not directly connected to the plasma membrane. Now I mentioned to you how if this is like a cell, let's say this is one cell, you have the nuclear membrane present here. Inside the nuclear membrane, you will have DNA, right? From the nuclear membrane, your rough endoplasmic reticulum will arise. I'm showing you like a 2D diagram. Of course, from this, the smooth endoplasmic reticulum will arise. From the rough endoplasmic reticulum, smooth endoplasmic reticulum will arise. Okay. Now, on the rough endoplasmic reticulum, you can find ribosomes being present. Why are they there? What is the use? Is the lumen only providing space for ribosomes to come and sit? Not really. If you think about it, let's say this is the endoplasmic reticulum's membrane. Inside the membrane of the ER, there is lumen. This is an aqueous compartment. Here you can find enzymes, certain proteins, certain ions and all of that. So, over here, if the ribosome is attached, okay, so let's say the ribosome is attached here. Once this ribosome synthesizes the proteins are synthesized in the form of a linear chain of amino acids. Many amino acids join to each other and they form a long chain. Okay. So when amino acids are like their long chain like that in their linear form, it's like a long chain, it cannot do its job. You know how many proteins are enzymes? Enzymes don't function unless they fit like lock and key with the substrate, right? So in this structure, they will not be able to do their job and they will need to be folded into three-dimensional structures. Crevices, pockets have to form. Only then they may possibly be able to function. These endoplasmic reticulum lumen will provide a safe space, a safe area for folding of proteins. You know how in proteins amino acids will have their own R group, side chains. Some of these R groups or side chains are hydrophilic, some of them are hydrophobic. If there are certain hydrophilic side chains present in this protein, if you just directly release it out into the cytoplasm, if they are hydrophobic, then the cytoplasm has a lot of water. It may not be able to fold very well. Its shape might get distorted and it may become useless. Okay. So what happens is, if this lumen of the endoplasmic reticulum, think of it like a tube. Imagine there's this ribosome sitting on top of this endoplasmic reticulum. The ribosome synthesizes the proteins and moves it, transports it into the lumen of the endoplasmic reticulum. In the endoplasmic reticulum, you will have several proteins called chaperones, like the ones that I've shown here in this diagram. Okay, so these are chaperones. What they do is they will provide a micro environment for the protein that will ensure its proper folding. If there are any side chains that are hydrophobic, they will fold in such a way that they are projected inside so that they don't have to come in contact with water. If there is any hydrophilic substances, they will be retained outside. So that way, based on what is the group or side chain present in the amino acid, they will provide an environment for the proper folding of the protein, and that happens very quickly inside endoplasmic reticulum. Outside it may happen very slowly, or outside it may not even happen. Protein can get aggregated and form clumps like that, which causes more harm than good to the cell. Okay. So inside the endoplasmic reticulum, it takes its proper shape very quickly. That is one way. One other way it helps in folding of proteins. Now talking about Golgi apparatus, Golgi is named after the scientist Camillo Golgi. Camillo Golgi is the person to have first observed Golgi structures inside a cell. Okay, so to honor him, for his contribution. We call those organelles Golgi apparatus or Golgi bodies. So, Golgi bodies are also membrane bound organelles. They are bound by a single membrane. They are made up of many flat disc shaped sacs. Imagine it in 3D. It's like many pouches that are folded like this, parallel to each other. Okay. So, they are called cisternae. These flattened sac-like structures that you find in Golgi, they are called cisternae, and they are pretty parallel to each other, and they measure between 0.5 to 1 micron in diameter. If you look at any cell, endoplasmic reticulum is very close to the nuclear membrane nucleus. Golgi apparatus is usually very close to the ER. They are all present very close to each other so that their functions can be coordinated. Okay. So these Golgi are concentrically arranged. I'll show you how it is. If this is like, let's say, one cell, animal cell, you have the nuclear membrane present here. Within the nucleus, you will of course have DNA. From the nucleus, the rough endoplasmic reticulum will arise. From the rough endoplasmic reticulum, the smooth endoplasmic reticulum will arise. Okay. On top of the rough endoplasmic reticulum, you have ribosomes present. Very close to the endoplasmic reticulum, you can find the presence of Golgi apparatus. What color do you think I should use? I've used a color, yellow maybe. So, in the Golgi apparatus, if you look at this image, okay, look here, this is an image 3D diagram of what happens inside a cell. Here is the nuclear membrane, here is the rough ER, here is the smooth ER, here is your Golgi apparatus, okay, very close to it. Notice how the sacs are not flat, they are like saucers. They are kind of bent, okay. So, because they are bent like that, here it is not shown very clearly, that's why I showed it to you in that diagram. The Golgi apparatus will have one face, okay, so towards this side, it is a convex face, and towards this side, it is kind of concave. The convex face is called the cis face or even forming face. The concave face that faces away from the endoplasmic reticulum's membrane is also called transphase or maturing phase. Now let's say in the cell there is requirement of a glycoprotein. What is a glycoprotein? It is a protein to which sugar is attached. You know that this protein part will get formed inside the ribosome. How does sugar get added to that glycoprotein? To add sugar, the protein will come to the Golgi apparatus. Let's say this ribosome synthesized a protein. It went into the endoplasmic reticulum. It got folded there. The endoplasmic reticulum got pinched off, okay, and sent this in the form of a vesicle to Golgi apparatus. Now, what has left the endoplasmic reticulum will come and fuse with the cis face of Golgi apparatus. So, that vesicle carrying the protein from ER will come and fuse with the membrane here, releasing that protein inside. Now, within the Golgi apparatus, that protein that was formed there, to that sugars will get added. it will get glycosylated. As it moves through the cisternae, it will get glycosylated. Once it comes towards the transphase, this protein along with the sugar will get pinched off from the Golgi apparatus and get released in the form of a Golgi vesicle. Okay, so once this gets pinched off, before it gets pinched off from the Golgi will add stickers to it. You know how from courier centers, different parcels will have to be delivered to different places. They put stickers on top of it and barcodes on top of it. That job happens at the Golgi apparatus. So it will put molecular tags on top of it, stickers on top of it, and based on the sticker, where it is supposed to get transported to, the vesicle will carry that particular protein to the designated location. If this glycoprotein was supposed to go and reach the plasma membrane, it will be transported there. It was supposed to go and get embedded into some other membrane, it will get transported there. Okay, same happens for glycolipids. If glycolipids have to form, lipids form in the smooth endoplasmic reticulum, but where does addition of sugar happen? It happens in the Golgi apparatus. It's called glycosylation. Okay, so that happens in the Golgi apparatus. Once it adds sugar to it, it will just pinch it off, and depending upon where it is supposed to go, it will add stickers and it will help it get transported to its destination. Okay, so these are some of the functions of Golgi apparatus. It not only packages the material, it even sorts out a lot of stuff inside the cell. Based on the where it is supposed to go, it can either be delivered to some place within the cell or even outside the cell, it can be secreted. Then, so whichever materials have to be packaged in the form of vesicles, they come from ER, come to the cis phase, fuse with the cis phase membrane, enter the cisternae. Cisternae is like the lumen of ER. So once they are inside the cisternae, whatever modifications need to happen, that will happen, and ultimately after they are modified, they will get pinched off as vesicles from the transphase and they will reach their respective targets. Okay, so Golgi apparatus is a very important site of formation of glycoproteins and glycolipids. Golgi itself does not synthesize proteins or lipids. The proteins will be synthesized by the ribosomes on ER. So that will be transported via a rough endoplasmic reticulum vesicle to Golgi. It only adds sugars to it and makes it a glycoprotein. The vesicles carrying lipids that arrive from the smooth endoplasmic reticulum, Golgi knows to add sugars to it. Golgi itself does not synthesize lipids or proteins. Okay, so that's about the functions of Golgi apparatus. Now I'm sure you understand why they are both part of the endomembrane system, because the output of one organelle is the input of another organelle. They communicate with each other for the formation of molecules like these. Okay, now let's talk about lysosomes. Lysosomes, as everybody knows, is the suicide bag of the cell. Suicide has a very negative meaning to it, but lysosomes are not negative organelles. Lysosomes are very, very essential. They make sure that if there is any waste in the cell, it is broken down in such a way that the broken down components can be utilized by the cell. Like it does very good kind of recycling inside the cell. So these are called suicide bags. They are bound by a single membrane. And within these lysosomes, hydrolytic enzymes will be present. Okay. How is this lysosome associated with the endomembrane system? We'll try and understand. I told you how Golgi will pinch off vesicles containing proteins or lipids. Okay. These hydrolytic enzymes that are present inside the lysosomes, they are proteinaceous in nature. What are hydrolytic enzymes first? In complex macromolecules, monomers or certain chemicals will be held together by means of chemical bonds, certain molecules. These will help it in breaking it down. Basically, they use a molecule of water to break bonds, hydro water, lysis breakdown. So they break down bonds by making use of water. That's why they're called hydrolytic enzymes. They're all proteins. And we know proteins are produced by ribosomes. So what basically happens during the synthesis of lysosome is the ribosomes that are present on top of endoplasmic reticulum will produce hydrolytic enzymes. They are pinched off as ER vesicle. They will come and combine or fuse with the cis face of Golgi. In the lumen or the cisternae of the Golgi, it will get modified. They will get assembled inside the cisternae, and it will pinch it off as a vesicle from the transphase. A vesicle that contains hydrolytic enzymes is just the lysosome. Lysosome is just like any other vesicle from Golgi. If it contains hydrolytic enzymes, then we call it a lysosome. Now, hydrolytic enzymes come in many different types depending upon the substrate it is involved in breaking down. If it breaks down nucleic acids, you call them nucleases. If it breaks down carbohydrates, you call it carbohydrases. If it breaks down proteins, then you call it proteases. If it breaks down lipids, you have lipases. So every kind of hydrolytic enzyme you can find inside of our lysosomes. And these hydrolytic enzymes are usually in their zymogen state, like enzyme precursor stage. Whenever there is acidic pH less than 5, then it will get activated and start breaking down things. It will not break down anything that is working well in the cell. If there is any organelle that is not working very efficiently, like a very old worn out mitochondrion, then it will break it down. If an amoeba has engulfed food, a vesicle is formed. It will fuse with that vesicle with the food vacuole and help in breaking it down. So that way this will help. Okay. Now, one question that you all might have: if a lysosome is bound by a membrane, lysosome has substances that are active at acidic pH. How is it that it does not break down the lipids present in its own membrane? I told you generally that in a plasma membrane, the sugar carbohydrates are present towards the outer side only. But in lysosomes, those sugars are present towards the inner side as well. Its structure is modified in such a way that the enzymes that are present in the lysosome will not be able to bind to it. If it cannot bind to it, it cannot break it down. Also, the high pH of lysosomes is maintained because in the membrane of the lysosome, you will find a lot of hydrogen pumps, proton pumps. They will keep pumping protons into the lysosome to maintain its low pH. More protons is less pH, right? Acidic. And what would happen if inside a cell one lysosome carrying such deadly hydrolytic enzymes bursts open? If it bursts, compared to the volume of the cytoplasm, the volume of the lysosome is very small. So the cytoplasm is almost neutral. Therefore, immediately the little bit of acidic nature of the lysosome will be neutralized by the huge volume of the cytoplasm. So it doesn't cause major harm to the cell. Okay, so that's about our lysosomes. Now let's talk about the vacuole. Vacuole is found only in plant cells. In plant cells, if this is the cell wall, inside the cell wall, you will have the cell, oh my god. Inside the cell wall, you will have the cell membrane, right? So at the center, usually you will find a big vacuole. The nucleus will somewhere be pushed to the side. Okay, so this is like the nucleus, let's say. This vacuole is bound by a single membrane that is called the tonoplast. Animal cells, very small cells, might have very, very small vacuoles, but this large vacuole in plants is a characteristic feature of plant cells. You don't find them in animal cells. Okay. Now, what's present inside the vacuole? Right within the vacuole, you can find water, some sap, some excretory substances, a whole lot of ions and waste substances will be present inside the vacuole. In plant cells, the vacuole sometimes can occupy as much as 90% of the volume of the cell. The tonoplast, this membrane will have many pumps present in it, through which ions that are present in the cytoplasm will be pumped into the vacuole against the concentration gradient. Okay, so the concentration of ions here is much higher compared to the concentration of ions in the cytoplasm. Okay, In plants, the tonoplast facilitates the transport of a number of ions and other materials against the concentration gradient into the vacuole. Therefore, the concentration of these ions is much higher inside the vacuole compared to the uh, presence or the concentration of ions in the cytoplasm. Now, there are two different types of vacuoles that you should be aware of. One is the contractile vacuole that you find in amoeba. That is not like this plant cells vacuole, that is different. And you can find food vacuoles in some protists. Okay, that is not like the plant cells vacuole, they perform different functions. Let's take example of amoeba only. So, if this is one amoeba, let's say this is one amoeba, it finds some food. What it does is, it will draw its pseudopodia around it like this. Visible, it will draw the pseudopodia around it like this, and eventually these membranes fuse and a vesicle forms within which food is present. This can be referred to as the food vacuole. Eventually, this food vacuole will fuse with a lysosome. Lysosomal enzymes will help in breaking down that food. Okay, so that is a food vacuole. About contractile vacuoles, you know, amoeba and many other protists are aquatic organisms. They are found in fresh water. They don't have a cell wall. The cytoplasm has water, but it has other dissolved solutes as well. So it's concentrated compared to the surrounding water. If too much water enters the cell because of endosmosis, because of concentration gradient, the cell could burst. So what these osmoregulatory vacuoles do, the contractile vacuoles do, is they will accumulate all the excess water inside the vacuole and they will pop. So the water gets released outside. Again, more water comes, it will all get collected inside the vacuole and then it will release it outside. It will just, you know, contract like this and release it outside. So that way it makes sure that the cell does not, you know, too much water does not enter inside the cell and make it burst. Okay. So that's about the vacuole. That's about the endomembrane system. In endomembrane system, whatever organelles were there, we've discussed that. Now I'm sure you can see a connection why these organelles are put together as the endomembrane system. ER and Golgi communicate a lot. The vesicles of ER will enter the Golgi and they will get modified there. Lysosomes are produced by Golgi membranes. Lysosomes are modified and packaged inside the Golgi, so that way there is a correlation. Vacuoles are also produced by membranes of the ER and Golgi. That way they are also connected. So that's about the endomembrane system. Now let's talk about the other organelles that we find in eukaryotic cells. Let's start with the mitochondria. Mitochondria is the plural form. Mitochondrion is singular. Mitochondria compared to chloroplasts are smaller, so you can't very easily see them or observe them under the microscope. How many mitochondria a cell has varies from cell to cell. Depending upon the physiological activity of that particular person, mitochondria, I'm sure everybody knows as the powerhouse of the cell. That's something that science students will never forget. So, why exactly is the mitochondria called the powerhouse of the cell? The reason it's called so is because within the mitochondria, large number of ATP will get generated. ATP is the energy currency. Whenever the cell needs energy for any metabolic activity, it will utilize energy from ATP. Since large amounts of that energy-rich molecule gets produced inside these organelles, we call them the powerhouses of the cell. Some of them have many mitochondria. If such cells are physiologically very active, like muscle cells in athletes, there will be too many mitochondria. If the cell is not physiologically very active, then the number of mitochondria there will be less. And its shape is usually sausage shaped or cylindrical. Size, the diameter is around 0.2 to 1 microns and the length is around 1 to 4.1 microns. Let's talk about its structure. The mitochondria has two membranes. Okay, I will show you a section of the mitochondria. Okay, so it's sausage shape. Let's take it like this, kind of cylindrical. This is the outer membrane. It is smooth. Inside the outer membrane, you have the inner mitochondrial membrane, which is enfolded like this. This is the inner mitochondrial membrane, which is convoluted, like it has many enfoldings. The space that is present between the outer and inner membranes over here, the space that is present between the outer and inner membranes over here, this space is called the perimitochondrial space or the intermembrane space. We'll come to this when we discuss the chapter respiration in plants. Okay, so this is the intermembrane space, and these enfoldings of the inner membrane of mitochondria is called, they are called cristae. Crista is singular, cristae is plural. Right within the inner membrane of the mitochondria, you can find a homogeneous substance present, similar to the cytoplasm, and that is called the matrix. It's kind of granular. Okay, so this is called the matrix. Inside the matrix, you can find the presence of circular DNA, similar to the DNA of prokaryotes. You can also find the presence of certain RNA molecules, and you can find the presence of ribosomes. But what's interesting about the ribosomes is that even though they are present inside the eukaryotic cell, these ribosomes are similar to the ribosomes of prokaryotic cells because they are of 70S type. Ribosome itself is an organelle. Now this ribosome is present inside another organelle, mitochondrion. So ribosomes here are called, can be called organelle within an organelle. If they ask you which organelle, which is an organelle within an organelle, you know it is ribosomes. Now let's talk about the cristae. Why do you think the cristae should be so enfolded? The enzymes and protein complexes, the complexes required for the generation of ATP, they are located in the inner membrane. Okay, so which color have we not used? Maybe black. So, I don't know, may not look very clean. Let me take pink only. So here, this is the location of those complexes involved in generation of ATP. More about it we learn in a chapter called respiration in plants. You have enzymes like ATP synthase, which is directly involved in the synthesis of ATP, and many other complexes are located in the inner membrane of the mitochondria. The more the surface area of the inner membrane, more such complexes can be accommodated, more ATP can be generated. In order to make sure that many such complexes are accommodated inside the inner membrane of the mitochondria, it is enfolded to form cristae. Okay? Now, let's talk about the next organelle, which is also bound by two membranes. It is found only in plants and certain photosynthetic eukaryotic unicellular organisms like euglenoids. And compared to the size of mitochondria, these are pretty large and they can relatively be seen under the microscope with more ease. The reason, these plastids are of many kinds. Depending upon which kind of pigment it bears, we classify them into three major categories. The first one is the most popular, chloroplast. The second one is chromoplast. The last one is leucoplast. Chloroplast contains many pigments, but predominantly it contains chlorophyll. Therefore, it is green in color. It imparts green color to plant parts. Colors other than green, like yellow, red, orange, all of these are because of the presence of chromoplasts. They impart colors other than green to different plant parts. Talking about leucoplasts, they do not have any pigments, so they are colorless. Leukocytes, WBCs, white color, no color, like they are pale, right? Very similarly, leuco means colorless. You can think about it like that. Leucoplasts are colorless plastids. They don't impart any color. They don't carry any pigments. Then why do they exist? They exist to store reserve food. Depending upon the nature of food these leucoplasts store, they are of three categories: alluroplast, amyloplast, elaioplast. Alluroplasts are leucoplasts that store proteins. Okay. Elaioplast stores oils and fats, lipids basically, and amyloplast stores starch or carbohydrates. Now, in your syllabus, you only have to learn about the structure of chloroplast in detail, not chromo or leucoplast. Let's take a look at the structure of chloroplast. It is mostly found in the mesophyll region of the leaves. If you don't know what mesophyll is yet, it's fine. We'll learn about that in another chapter later. They are cells found in the leaf which are majorly responsible for photosynthesis. The shape of the chloroplast is variable. In some algae, it can be ribbon shaped, it can be cup shaped, it can be plate like, it can be cylindrical, it can be lens shaped. Its shape varies. The size range in length is about 5 to 10 micron and in width it's about 2 to 4 micron. The number of chloroplasts in a cell also varies. In unicellular algae like Chlamydomonas, there's only one chloroplast per cell, whereas in the mesophyll cell of higher plants, you can find as many as 20 to 40, sometimes even 60 chloroplasts per cell. Now let's talk about its ultrastructure. A chloroplast, like I mentioned, is made up of two distinct membranes: an outer membrane, which is smooth, and an inner membrane, which is also smooth. There are no infoldings or cristae in this case. This is the outer membrane, and right inside that you find the inner membrane, and the space that exists between these two membranes is called the periplastid space or the intermembrane space. Inside the two membranes lies a granular matrix similar to the matrix of mitochondria, but it's found with a different set of enzymes. This is called stroma. The pigments that I spoke to you about, the chloroplast, the chlorophyll, carotenoids and all of that, they are located inside the membrane. It's not located inside the outer membrane or the inner membrane. Within the inner membrane also there are more membranous structures that you can find. So they are tiny flat sac-like structures that stack one on top of each other like that. Each single sac is called a thylakoid, and that entire stack is called granum singular. Grana is plural. Many such grana form inside the chloroplast. These grana are linked to each other by means of membranes again that are called stroma lamellae. Okay? It is within these membranes that pigments are present. Pigments are present in the form of complexes. More about how they are present we will learn in the chapter photosynthesis. Now, in the stroma of chloroplasts also, you can find the presence of circular DNA, ribosomes of 70S type, and some RNA as well. Because these two organelles, chloroplast and mitochondria, have DNA of their own, they have ribosomes, they have RNA, they can make some of the enzymes that they require to function. They are not fully dependent on the DNA present inside the nucleus. These two organelles are said to be semi-autonomous. They are somewhat partially self-sufficient, you can say. Okay, so they are said to be semi-autonomous. Now that I'm talking about pigments, I want to give you one important information. The lipids that I spoke about just now, like chlorophyll, carotenoids, the pigments that are responsible for imparting green color, orange and red colors, they are usually lipid soluble pigments. Since these membranes are made up of phospholipids, they dissolve in them and they are present within those membranes. If you ever touch a leaf, it's not like the green color in the leaf will get transferred to your hand. If you touch an apple's peel or a carrot, the red and orange colors will not get transferred to your hand. As opposed to that, if you touch a beetroot, if you squish a pomegranate seed, or if you squish a blueberry, color is produced, right? Purple, blue colors, those pigments will get released, okay? And your hand will also, the color will get transferred to your hand also. So if you notice why that happens, that's because those pigments that are responsible for your purple color, pink color and blue color, they are not lipid soluble. They are water soluble. And you can find them located within the vacuoles. Okay, so some pigments are located inside the vacuole. So they are water soluble. That's why when you touch them, since they are water soluble, it gets transferred. Okay. Here, if only the chlorophyll also was water soluble and it was present in the vacuole, then just one time if it rains, all of the pigment chlorophyll that is present inside the leaf would have leaked out, right? So that's why it's not water soluble, it is lipid soluble and it is present located within these thylakoid membranes inside the chloroplast. Now let's move on and talk about another organelle that is bound by two membranes. I mentioned in the beginning of the class itself that the nucleus was first discovered by Robert Brown in the year 1831. Later, Fleming, not Alexander Fleming, this is a different Fleming, he used basic dyes. Like basic dyes means stain to visualize the cell, we use that is basic in nature. Since the nucleus contains DNA, deoxyribonucleic acid, since it's an acid, basic stains, if you add to it, it will pick up color, it will impart color and you can visualize that under the microscope. So he used basic dyes and he gave the name chromatin to the material of the nucleus, basically the DNA. Chroma means color, okay. So the nucleus, when the cell is not dividing, we say that the cell is an interface. Interface nucleus, DNA will exist in the form of long threads, that is called chromatin. In an interface nucleus, you can see chromatin. The chromatin is not just made up of DNA, it is made up of DNA and certain proteins called histone proteins and many other non-histone proteins also. So you can say that it is a nucleoprotein, nucleic acid, DNA plus proteins. Apart from that, you can find the nuclear matrix, which is similar to the cytoplasm kind of in composition. And you can find one-to-many nucleoli present within the nucleus. The nucleoli are not membrane-bound structures. Okay, they are non-membrane bound, but they pick up stain inside the nucleus and they are visualized as spots inside the nucleus. Yeah, so they are spherical bodies carrying out photosynthesis. Why will nuclei perform photosynthesis? No. They will not perform photosynthesis. They will synthesize a type of RNA called ribosomal RNA, carrying out synthesis of rRNA. That is the function of nucleoli. Okay. Now, let's talk about the structure of nuclear membrane. I told you that nucleus also is bound by two membranes. There is an outer membrane and inner membrane that are parallel to each other, as you can see in this illustration. This is the outer membrane, this is the inner membrane. But you know what? The two membranes are not like continuous. If this is one membrane and this is the other membrane, you know, in some places, it's a little smaller so you have more clarity. What happens is these membranes themselves will fold upon itself in some regions. It gets folded upon itself like this in some regions and form pores that are called nuclear pores. So basically, when I say there are two membranes, you have to visualize it like this. This is one membrane. This is another membrane. Okay, so there are two such membranes. These two membranes have folded upon itself. That means they are like this right now. So if they folded upon themselves and formed this kind of a structure, if this side also it's like that, in between there will be a pore. That is how it is. There is space that exists between the two membranes of the nuclei, and that is called the perinuclear space, that measures between 10 to 50 nanometers. Okay. The outer membrane, from the outer membrane, the rough endoplasmic reticulum will usually start originating. Similar to how the rough endoplasmic reticulum has ribosomes studded on top of it, the outer membrane also can show the presence of ribosomes. That can also be associated with ribosomes. Yeah, like I mentioned, at many places in the nuclear membrane, you can find nuclear pores. Nuclear pores are regions through which substances can get transported from the nucleus to the cytoplasm, from the cytoplasm to the nucleus. Okay, it could be RNA, small proteins, they can get transported in both directions. Okay, what will get transported? RNA as well as protein. Why does it have to get transported? You will understand in another chapter called molecular basis of inheritance. Okay, now normally there's only going to be one nucleus per cell, but sometimes in some cells nucleus may be entirely absent, as in the case of mature RBCs. They are enucleated. While they mature, they lose their nucleus. Sieve tube elements in angiosperm phloem, they don't have nuclei. So some cells can completely lack a nucleus. There are some cells which can be multinucleate. They can have more than one cell. Okay, for example, if you take the prenuclear endosperm that has many nuclei. If you take tapetum cells in anther, they have multiple nuclei. So it's possible in some cases where the cell does not have even one nucleus, or it has more than one nucleus, but the general norm is that usually in most cases one cell will have only one nucleus. Okay, now inside the nucleus, DNA will be present. The DNA can exist in different forms. When the cell is not dividing, it usually exists in the form of what is called chromatin fiber. But once the cell decides to undergo cell division, the DNA will become more compact, it becomes shorter and thicker, and it forms what is called the chromosome. This is the structure of a chromosome. A chromosome after replication will have two chromatids present. This is one chromatid. This is the other chromatid. Whatever information is there, same thing will be copied onto this side as well. Okay, so these are the two chromatids. You can see that they are not uniformly thick. There is a region where they are narrow. This narrow region is called the primary constriction. Constriction means becoming narrower. Primary constriction or the centromere. On either sides of the centromere, you can find proteinaceous disc-like structures called kinetochores. They basically help separate the chromatids during cell division, more about which you'll learn in the upcoming chapter. Okay. Now I mentioned that this primary constriction is called the centromere. That does not mean it necessarily have to be at the center at all times. The position of the centromere can vary. If it is present in the center, like in this case, it is called a metacentric chromosome. If it is present a little away from the center, then you call it submetacentric chromosome. If it's present too much away from the center, more close towards the tip, Then you call it acrocentric chromosome if it's present at the terminal end, absolute tip, then you call it telocentric chromosome. Some chromosomes, in addition to having this primary constriction, will also have secondary constrictions. Some of them, not all of them. Another narrow region. That secondary constriction, beyond that whatever chromosomal part tip will exist, no, that is called the satellite. It's usually the DNA that is present at the satellite region which will form the nucleolus later. Over there, you will have genes that are responsible for the synthesis of ribosomal RNA. Okay? Now, here in metacentric chromosome, you can see that both the arms, like this is one chromatid, this is the second chromatid. This chromatid has one arm, two arm, this has one arm, two arm. Totally four arms are there. In metacentric, submetacentric, acrocentric chromosomes, you will have four arms and two chromatids after replication. In telocentric chromosomes, you will have two chromatids and two arms only because centromere is present at the tip. Whenever there are two arms and the two arms are not, whenever there are four arms, okay, like this, or whenever there are two arms per chromatid and they are not equal, here it is equal, both of them are similar, right, not too much difference, then there's no problem. In these two cases, in submetacentric and acrocentric chromosomes, you will have one long arm and one short arm. The short arm is called the P arm. The long arm is called the Q arm. P for petite. Petite means small, right? It is smaller, so it's called the P arm. The longer one is called the Q arm. That does not exist here, P and Q, because both are equal. Here, there's only one arm, so there's no P and Q here as well. Now let's talk about ribosomes, which are non-membrane bound organelles, universal organelle, organelle within organelle. These are granular structures that you can find in the cytoplasm, on the nuclear membrane, on the endoplasmic reticulum, inside the mitochondria, inside the chloroplast, right? So in multiple locations inside a eukaryotic cell, you can find ribosomes. These ribosomes were first described as dense particles by a person called George Palade. That is why the ribosomes are also called Palade particles. Write it in capital letters, the name of a person. He discovered them in the year 1953. What is the ribosome made up of? Ribosome does not have a membrane, no phospholipid bilayer, nothing. What is it composed of if it does not have a membrane? It is made up of only two components. It is made up of a type of RNA called ribosomal RNA, rRNA. And it is made up of certain proteins. There are many different types of ribosomal RNA, single-stranded. So there will be many types of ribosomal RNA like that. And associated with that, you will have certain proteins present too. That itself will form the structure of ribosome. Similar to how in prokaryotes ribosomes existed as two subunits, even in eukaryotes, it exists as two subunits. The large subunit is 70S. Wait. The large subunit is 60S and the small subunit is 40S, but together they are not 100S, they are 80S. The prokaryotic ribosome you already know, 50 plus 30, it's not 80, it is 70, I've told you that. Now, prokaryotic cells always will have 70S ribosomes only. But in eukaryotic cells, you find both 70S ribosomes as well as 80S ribosomes. The ribosomes that are present on, the ribosomes that are present inside the mitochondria, the ribosomes that are present inside the chloroplast, they are 70S type even though they are present in eukaryotic cells. The ribosomes that are present in the cytoplasm, on the nuclear membrane, on the rough endoplasmic reticulum, they are 80S type. Okay. So, that's about ribosomes. Their function is to synthesize proteins. I've told you that already. Now let's learn about the cytoskeleton. Cyton means what? Cyton means cell. Skeleton you already know. The framework of our body, that is our skeleton, bones and cartilage it's made up of. Similar to how we have a skeletal system inside our body that gives it shape, it gives it mechanical support, right? Very similarly, the cells also have a cytoskeleton of its own, where, like I mentioned earlier, it's not made up of bones and cartilage, it's made up of proteins. That is called the cytoskeleton. That is what will help in maintaining the shape of the cell to an extent. It is of three types. You have microtubules made up of a protein called tubulin. You have microfilament that is made up of another type of protein called actin. I'm sure everybody knows actin, right? You would have learnt about it in sliding filament theory of muscles. Then you have intermediate filaments that are made up of different types of proteins such as keratin, etc. Okay. What is their function? The same function that the skeleton has in our body. It provides us mechanical support. If not for our skeleton, we couldn't stand erect like this. We would be a blob of flesh lying on the ground. It provides mechanical support and makes sure that we can stand erect. Likewise, the cell also gets mechanical support because of its skeleton. It helps in motility. Even in us, movement is possible because of our skeletal system and our skeletal muscles, right? Otherwise, I couldn't be able to move my jaw today. We wouldn't be able to blink. We wouldn't be, not blink, maybe blink does not involve bones. Yeah, so basically I can't move my hand like this. You can't take down your notes. If not for our skeletal system, we can't move from one place to another. So it helps in motility and movement as well. Same for cells, okay? And then it helps in maintaining the shape of the cell, okay? Now, this is one of the most overlooked parts of the cell, but they play such an important role in the functioning of the cell. Whenever we say that vesicle from Golgi apparatus gets transported to the plasma membrane, vesicle from the ER gets transported to the Golgi, I usually, when I was in your age, I used to assume that the vesicle will swim in the cytoplasm and go reach there. But that's not actually how it works. These cytoskeletal elements will form roadways inside the cell, okay? So it is on these roadways that the vesicles will get transported. That way it will help in transporting of vesicles. It will provide a pathway for the vesicles to get transported. There will be proteins called motor proteins which will literally carry the vesicles on its back, slowly carried on top of the cytoskeletal elements and deliver it to its targets. Okay, and also I mentioned that these are like roadways. It's not like permanent roads are built inside the cell. Depending upon the need of the cell, the roadways can get broken down, they can reform, and a whole lot of reorganization can happen inside the cell. Okay, so they are very dynamic structures. This is an image from fluorescent microscopy that I've taken of the cell, because very rarely you see cytoskeletal elements represented inside a cell. You will see all organelles labeled, but cytoskeletal elements not many textbooks show. So this is just cytoskeletal elements that you can see. You can see how it is spread across throughout the cell. In the center, that black area is where the nucleus was possibly present. So the blue color things are the microfilaments. They are spanning across the entire cell. Then you have microtubules represented in green color and intermediate filaments are represented in red color. Now, here's what it looks like inside a cell. You can see microtubules are tubular. They are like tube sort of things. We learn about these tubes later when we are discussing flagella, cilia and centriole also. Then you have microfilaments which are also called actin filaments. About its structure you will learn in the chapter locomotion and movement. They are globular G-actin. They form a filamentous F-actin and they wind around each other like that. That is the actin filament. And then you have intermediate filaments made up of many different types of proteins. Here is how it is spread across inside a cell. You can see how they are connected to different organelles as well. You can see this orange colored intermediate filament is forming an intermediate structure between different organelles, from plasma membrane to the ER, from ER to the mitochondria. So they are keeping things in place. Okay. Now, in this image also, you can see the presence of cytoskeletal elements. To show you, I have gotten this image. All of these structures that you see here, these green color tubular structures, they are tubulin proteins, microtubules. Okay. So here also you can see the presence of cytoskeletal elements. Yeah. Now let's talk about centrosomes or centrioles, which are organelles that are found only in animal cells. You don't find them in plant cells. And similar to ribosomes, these are also not bound by a membrane. They are non-membrane bound organelles. The centrosome, one centrosome will contain two cylindrical centrioles that are perpendicular to each other. They're always like an L. They're like this, perpendicular to each other. Surrounding these two centrioles, there is an amorphous pericentriolar material that you can find. Usually, the centrioles are located very close to the nucleus of animal cells. Per animal cell, you will usually have only one pair of centrioles or one centrosome. It is involved in formation and maintenance of spindle fibers, organization of spindle fibers during cell division. How exactly it works, I will tell you when we are discussing the chapter cell cycle and cell division later. So now just understand that is their role. So the structure of the centriole is like a cartwheel, they say. You know a cartwheel, right? Like a bullock cart's wheel, it has like an outer circular framework and there are many spokes that are radiating from the center. Its structure, if you look at it closely, it will look very similar to the structure of a cartwheel. If this is one centriole, you're taking one centriole, looking at it from above or taking a thin section and looking at it from above, here's what it's going to look like. You can see there are many circular things here. Each circle is a tube actually. Okay, so that tube is a microtubule. Centrioles are composed of microtubules, the same microtubules that we saw in the cytoskeletal elements. Okay, they are tubular. Micro means small. Small tubes is called microtubule. It's made up of a protein called tubulin protein. So over here, you can find 1, 2, 3. Three microtubules together. That is called a triplet. How many such triplets do we have? We have 9 such triplets. Let's start from here clockwise direction. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets that are evenly spaced on the periphery, on the outside. Okay. So these peripheral triplets are made up of tubulin protein. Basically, they are microtubules. Now, the triplet here is linked to the adjacent neighboring triplets on either sides by means of what is known as linkers. They are linking the adjacent triplets, so obviously they will be called linkers. At the center of the microtubule, you will find a proteinaceous structure that is called the hub. From the proteinaceous structure, again, many other proteins will start arising and they will join with one of the 3 triplets here. One of the 3 microtubules in the triplet here. And since they appear similar to the spokes, they are called radial spokes. If it's a circle, how do you draw a radius to a circle? From the center to the periphery. These are like that, right? So these are called radial spokes. Since there are 9 triplets, there will be 9 radial spokes in each centriole. You have 2 such centrioles that are arranged very parallel to each other. Now, in cilia and flagella of eukaryotes also you have a basal body, but that's nothing like the basal body that you find in prokaryotes. The basal body in eukaryotes for cilia and flagella, locomotory structures, is composed of centriole-like structure. Okay. Apart from that, it also, I mentioned, right, it organizes the spindle fibers during cell division. More about it we'll learn in another chapter. So here's what it looks like. Now let's talk about, I think this slide is repeated. I just spoke about it, right? Now let's talk about the locomotory structure, cilia and flagella. Cilia and flagella are both extremely thin hair-like outgrowths that arise from the surface of the cell in eukaryotes. Both of them have the same function. They are involved in locomotion. So it's movement. It's involved in locomotion. Usually cilia are shorter and more in number. Flagella are longer and more in number. The cilia, there'll be many cilia. They work like oars. Like you've seen that boat race in Kerala where many people are sitting and they're rowing their boats. As and when they're rowing the boats in unison, the boat gets propelled forward, right? So the cilia also beat like that, and when the cilia is beating, the organism is pushed forward. The cilia can either cause the movement of the organism itself, or like the ciliated epithelium present in our respiratory tract, they beat. When they beat, the surrounding fluid will move. There are ciliated cells present in the lining of our fallopian tubes. They also beat and move the, egg, right? Egg or the blastocyst, whatever. So the cilia can make the cell itself move, or it can make the surrounding fluid or the surrounding substance move. So when you look at the electron microscopic image of both cilia and flagella, the structure will appear very similar, and in both cases there is a central core called the axoneme that is surrounded by a plasma membrane. Okay, so this is the flagellum. Same structure will be present in the cilia as well. Outside yellow color, what you're looking at is the plasma membrane. Inside that, what you're looking at, this green color structure, that is the core of the cilia or flagella, which is referred to as the axoneme. So here, the core, like I mentioned, is called the axoneme. You can notice how outside it is covered by the plasma membrane. Okay. This is the plasma membrane. Inside that, again, microtubules are present. Many microtubules are present. Small tubes made up of tubulin protein, alpha and beta tubulin. More about that we'll learn later. Now, you can think of this as that clicking pen that you get. You know, some pens are multicolored, like you have red refill, green ink refill, blue ink refill, pink ink refill. So you just have to click it and then the color will change, right? So you can think about it like that. Outside there is plasma membrane. Inside that there are many tiny tubes present in jodi, like doublets in pairs. Okay, so there are 9 such doublets present on the outside. Again, this is, if this is the flagella, I'm looking at it from above. I've taken a thin section, put it under the microscope, and then I'm looking at it from above. Each circle that you look at here is one tube. Okay, one tiny tube. How we saw in centrioles, there were 9 triplets. Here you can see that there are 9 doublets. There's 1 and 2. In each doublet, there are 9 such doublets. 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 doublets are present peripherally. At the center, you can find 2 singlets. This is one, this is one. There are 2 singlets at the center. This is referred to as 9+2 array or 9+2 arrangement of microtubules. In centriole, at the center, you did not have microtubules. You had a proteinaceous hub, but they were not microtubules. Over here, peripherally also you have microtubule. At the center also you have microtubule. Okay. So the axoneme usually has 9 doublets peripherally, and centrally there are 2 microtubules. The central tubules are connected by means of bridges. You can see how there is a bridge that is connecting the 2 central microtubules. Around the central microtubules, you can also find a sheath, which is called the central sheath, because obviously it's present in the center. From the central sheath, proteins will arise and it will get connected to one of the 2 microtubules in the doublet, and that also forms radial spokes. Since there are 9 doublets here, over here also there will be 9 radial spokes. The doublets are connected to the neighboring doublets by means of linkers over here as well. That is the structure of flagella and cilia in eukaryotes. It's mostly made up of tubulin protein. As opposed to this, do you realize and recollect how simple the structure of prokaryotic flagella was? It had just 3 components, a very simple sort of a basal body, a hook and a filament. But here the structure is far more complex. Now let's talk about microbodies. Microbodies are some of the smallest vesicles that you can find inside both plant cells as well as animal cells. These microbodies usually contain enzymes, different kinds of enzymes. One such microbody is peroxisomes. Peroxisomes are known specifically to contain a type of enzyme called catalase. So this catalase basically breaks down hydrogen peroxide into water. It converts it into water and makes it less toxic. So like that, you will have many different types of enzyme containing vesicles present in the cytoplasm of plant cells and animal cells. We call them microbodies. Micro, very tiny vesicles. That's why they're called microbodies. So with that, we have pretty much completed this entire chapter. We started off with the cell theory, the contribution of various scientists, and we spoke about the prokaryotic cells, its structure, its function in detail. Then we discussed the eukaryotic cell, its different organelles, their structure, their function. And by now, I'm sure everybody has mastered the chapter cell, the unit of life, and I'm sure you will be able to solve the questions with ease. Very soon, I will also bring to you questions for practice. We can solve them live. And yeah, that's about it. I hope this session was interesting, engaging, and informative. I'll meet you again in the next lecture with another chapter. Until then, take care of yourselves, stay healthy, stay happy, and keep studying hard. Thank you.